Sound check, students. Hello, good morning, each and every one of you. Good morning, students. Good morning. Students, quickly tell me in the chat right now if I'm audible and visible to one of you students. Good morning. How was everyone doing, students? Good morning, each and every. Right. Students, today is going to be again, I know what happened last time, right? Last time what happened was there was a major issue with the voice, major issue with the entire stream getting end at right. And we decided that, just because the stream got ended, right, we wanted to do the session one more time, but the issue was not solved. Yesterday also we came to the office and yesterday also we tried. And every single time we switch on the stream, it was ending automatically. There was a network issue. So I hope everything is resolved right now. If I'm properly audible, students, quickly some fire emoji in the chat if I'm properly audible. Because we do not want, again, the same thing to happen, students. Is it fine now, students? Is everything fine right now, today's class? Good morning, students. Now, okay, students, today I'll be teaching we will be doing the entire marathon today, right? We will try to finish every chapter in 1 hour, right? Because we have almost chapters, a lot of time to finish all of them, right? If you do entire thing, it's going to take whole day, right? So we'll be doing something called a summary, right? Right now, in today morning class, I will be teaching you the concepts. No question practice. Only pure concepts where you can go wrong, where you do not understand something. Then next class is going to be in the evening where we'll be doing purely question practice. So morning class is going to be, is it repeating again? No, I don't think so, right? Right. So again in the evening class we'll be doing the question practice. So all of you are set, learn the concept right now, and in the evening class all you need is question practice and you're good to go. Students, with that same energy, can we start our students? Can I get some likes in the chat, students? Can I get some likes in the chat, all of you? Fire emoji in the chat, students. I have tried so much. Again, oh my god. Students, am I audible right now? Tell me quickly in the chat, all of you. Am I properly audible now? Again, tell me. Now you tell me. I don't think so. It's repeating again now. I don't think so. All of you just trying to scam me at this point. Yeah. Right? Here now, students. Quickly in the chat. Now, students, I'll be asking you a lot of questions. So keep answering the chat. So I want to keep a track of everything. Okay. Now, first of all, the first chapter is going to be sexual reproduction of flowering plants. You cannot ignore this chapter. It has very, very high weightage. Clear, students? Now, first of all, students, when you think about a flower, what comes to your mind? Can anyone tell me in the chat, students? When you think about a flower, what comes to your mind? For me, flower always means happiness. Right now, every single time I see a flower, it instinctively makes me happy. Now, but if you are a scientist, if you are a botanist, right, like me, right, apart from looking at a flower at beautiful and everything, you will know that a flower is actually a reproductive organ. Yes, it is a reproductive organ. In fact, students, if you look at a typical flower, it's a flowering plant flower, right? Here it has something called as 4 whorls, 4 different whorls. Right, 4 different whorls. Here we have something called as the outermost whorl. Now imagine 1, 2, 3 and 4. Here the outermost whorl is called as the calyx. And all of you see here, the outermost whorl is called as the calyx. And yes, in some cases, the calyx can be photosynthetic in nature. Now, the second whorl is called as the corolla, which is mainly made up of petals. It is mainly made up of petals in general. That is the corolla. Now, students, can anyone tell me in the chat, do every single flower in the world has colorful petals, or do we find certain flowers where we do not need to be colorful? In the chat, do you think always flowers are supposed to be beautiful? All the flowers will be beautiful. That is not really the case. Most of the flowers which are colorful are generally insect pollinated. All of you understood? If it is not insect pollinated, it doesn't have to be colorful. There are some people, no, they don't care. See, for example, me only. Sometimes when I go out to the mall, now I don't care. I go in my chappal. I go wear chappal, I wear my shorts and I go to mall now. I don't care anymore. Right? So if you're like me, you don't care a certain thing. There are flowers which you don't care. They don't care at all. Right? Then we have the third whorl that is called as androecium. Now Understand the difference. What is the difference between androecium and stamen? Collection of a stamen or a group of stamen is called as androecium. And a stamen has two main parts. Can anyone tell me? A stamen has two main parts. The first part is called as the long slender filament. Can all of you see? Long slender filament and a bilobed anther. And a bilobed anther and a long filament. Then we have the innermost wall, that is gynoecium. And gynoecium is nothing but a collection of a what? Pistil. Collection of pistil. And if you can see, students, it has three main parts here. Now, uppermost part is called as a stigma. Now, students, imagine stigma as a, where helicopter will come and land, no? We have a helipad, where helicopter comes, it comes and it lands here. That is called as the stigma, landing platform. Then we have a long style and the basal part is called as the ovary. Now, again, students, is the ovary actually the gamete here? Ovary is not the gamete. Inside the ovary, we get the ovule. Ovules. Is the ovule gamete again, students? Is the ovule gamete in the chart, students? Do you think the ovule is the gamete right now? No. Inside the ovule, there is something called as the embryo sac. Do you think embryo sac is the gamete right now, students? Embryo sac also is not the gamete. So please be clear on what exactly is the gamete here. It is not the ovary, not the ovule, not the embryo. Yep. Let's read a few lines of NCERT and let's get some concept clear. Right. Now, androecium consists of whorl of stamen. All of you know it consists of stamen representing the male reproductive organ, and the gynoecium represents the female reproductive organ. So gynoecium is female, androecium is male. Now, the two parts of typical stamen are long slender stalk called as filament, long slender filament, and a bilobed anther. A bilobed. Now, why do we call it as bilobed? Some of you might ask me, sir, why do we even call it bilobed structure? Are there any two lobes present here? Like your brain, no? Lobes. Similar to our brain, there is something called as bilobed anther. Can all of you see here, students, the diagram? Here, if you closely look at it, this entire sectioning, right? This half, this side is called as lobe 1. This other side is called as lobe. So, inside a typical anther, if you take a cross section, it has one lobe on this side, one lobe on the other side. That is why it's called as bilobe. Now, students, now there is something called as theca here. What is theca? Theca is nothing but a cavity. Theca is nothing but a small cavity. Inside a small cavity, inside that we have microsporangium. Understand? One lobe has two theca. So, can I call it as a dithecous? Yes, so students in the chat. Can I call it as a dithecous structure now? Dithecous. Okay. Now, students, can anyone tell me how many thecas are total? In total, there are totally, students, 4 thecas here. 1, 2, 4. So, can I also call it as tetratheca? Some tetratheca, but we don't say tetratheca because we always generally consider one part of lobe. So, we call it as ditheca. Now, I told you, students, this theca is nothing a small cavity. Like, imagine a small cavity here. A small cavity. Inside this cavity, there is something. Inside the cavity, we have micro. Okay. Now you tell me, students, there are 4 theca. Every theca has microsporangia. Okay. So how many sporangia, microsporangia has to be there in total? There are in total 4 sporangia. So we can also call it as tetrasporangiate. Okay. So because there are 4 positions where sporangia is present, I will call it as tetrasporangiate structure also. Understood all of you? Tetrasporangiate structure. Okay, done. Now, students, if you look at one, I told you, this entire structure is called as a microsporangia. Microsporangium. Okay. Now, if you look at this one microsporangium present in one theca, it actually has 4 walls. Now, can anyone tell me, students, which is the wall here which provides nutrients? Anyone? So, if you look at this is a structure of a microsporangia. It has 4 walls here. 1, 2. Here the outermost wall is called as your epidermis. Second layer is called as endoderm. Third layer is called as middle layer. And the innermost layer here, the innermost layer is called as the tapetum. Now again, students, remember very carefully, it is a 2-mark question. Here what happens is, the first 3 layers are there, right? First 3 layers, their main function is protection and dehiscence of the pollen grains. While the innermost layer called as your tapetum, the main function is nutrition. Apart from nutrition, friends, tapetum also produces pollen kit and also something called as your callase enzyme. And I will tell you where you encounter this callase enzyme later on. Okay. Now, students, that is your microsporangia. Now, I can ask you one more question, students. No voice breaking, sis? No voice breaking again? No, right? Listen to me. Now, apart from, can anyone tell me what is present inside a microsporangium? I told you, this is the entire anther, one theca. Inside one theca, we have one microsporangium. But inside the microsporangium, what do we? Is my voice breaking still again? My phone once. One of you? Then something's wrong with the studio. We have to get it fixed. Yesterday we spent so much of time on this, but it's happening. Unlucky black magic someone has done. Students, pray for a studio once again. All of you pray for the studio. What do we get inside a microsporangium? This inner part is there, right? Inside microsporangium has something called as sporogenesis, and this sporogenesis itself develops into the spore and pollen grain. Listen to me now. Okay, all of you. Very simple. The chapter is very simple. Now I will teach you something called epidermis, endothelium, middle layer, tapetum. Okay, tapetum is going to be nutrition function, right? Nutrition. Now, students, I will tell you the formation of. Now I will teach you the formation of male gametophyte. Can we start, students? Can we do the formation of your male gametophyte? Now, before we start, can anyone tell me what is the male gametophyte here? Is the male gametophyte the microsporangium, students? Is the male gametophyte the entire anther, students? What is the male gametophyte? Can anyone tell me what is the male gametophyte here? Breaking up. Oh my god. Oh my god. Today, please pray. One namaskar. Please pray. Everyone says some magic you do. How much ever we do, it's going to be the pollen grain. Okay. Now, students, now I will teach you something called as the microsporogenesis. It's a very famous question. So listen very carefully. Okay. What exactly happens here? Okay. What exactly happens here? Now, microsporogenesis, where does it start from? Microsporogenesis starts from a cell which is diploid in nature, that is called as spore cells. Now, students, one of the sporogenous cell becomes very large and we call the sporogenous cells. Okay. I will keep this thing inside my mouth only now. Sporogenous cell, one which becomes large, is called as microspore mother cell. And this microspore mother cell is again diploid in nature. Now, students, you tell me which process is called as reduction division. The chat. What is called as reduction division? Here there is a reduction division. Reduction division here is called as your meiosis. So, one cell to two cell, meiosis 1. So, here you can see diploid to haploid, then meiosis 2, and we get 4 cells, and this arrangement is called as microspore tetra. Is called as microspore. Okay. Please, God bless the studio students. God bless. Called as microspore tetrad. Yeah, microspore tetrad. Now, students, understand here. Microspore tetrad has some layer around it. This layer is called as callose layer, and it does not break easily. So we need an enzyme here. Now, can anyone remind me again from where do we get this callase enzyme? Can anyone remind me from where do we get the callase enzyme? I'll tell you, students. Mahalakshmi, don't. I'll tell you. Okay, this callase enzyme is produced in the tapetum itself. Tapetum. Okay. Now listen to me, all of you, very carefully. Now we finally get the microspore. Now, are these microspores the gametes or the male gametophyte, students? Can anyone tell me, are these microspores male gametophyte? No, they are not the male gametophyte. Okay, because this microspores now undergoes something called as vacuole formation, and we get a large cell, and finally after mitosis, asymmetrical mitosis, we get our male gametophyte. We get our male gametophyte. And this male gametophyte right here is the pollen grain. Now someone was asking me, sir, what is the difference between a gamete and a gametophyte? This entire structure is called as gametophyte. Gametophyte. But inside this gametophyte, you see a cell here called as generative cell here, all of you. This generative cell will divide into gametes. So gametes is different, gametophyte is different. Gametophyte is different. So Calais enzyme, very simple students, understand. Here this entire structure is held together, like there is a casing there. Now this casing has to break open to make the microspores free. The freeing is done by Calais enzyme. Okay. Now student, this is your entire pollen grain. Now here understand students, there are two types of cells. They can ask you the diagram. So easy 2-mark question where they can ask you the diagram here. So draw this diagram and then you need to write it has two cells. One is called as the vegetative cell. Now all of you tell me students, this vegetative cell, is the shape proper or irregular in nature? The shape right here is irregular in nature. Irregular shape. Then we have a small cell called as generative. In fact, this generative cell will float in the cytoplasm of your vegetable. Very tiny, tiny. Okay. Now understand students. This entire pollen grain has an outer layer called as exine. So this exine is made up of a very, very tough, very highly resistant material called as sporopollen. The exine is made up of sporopollen and inner part is called as the intine. And the intine is made up of cellulose and pectin. Again, it's a 1-mark or 2-mark question. Cellulose and pectin. Okay, now students. Now in this entire layer of your exine, right, there is sporopollen. But at a very small point, sporopollen is absent. That small point where sporopollen is absent, that is called as what students? That is called as germ pore. There is a small area where sporopollen is absent, that is called as germ pore. Now some of you might ask me, sir, what is the function of a germ pore? Very simple. The main function of germ pore is that from the small hole, right, the pollen tube will come out. Pollen tube. Okay, pollen tube comes out. Understand? Okay. All of you understand sporopollen, cellulose and pectin present in the intine, and when the pollen grain is mature, it contains two cells, vegetative and generative. Now students, all of you tell me in the chat, this particular pollen grain has how many cells right now? It has two cells. So this is called as two-celled stage. It is called as what? Two-celled stage. Now listen to me. What will happen if these two cells divide into two more cells? This cell will divide into two more cells. In such case, we have how many cells? 1, 2. Such case, we have 3. There is something called as two-celled stage and three-celled stage. And listen to me students, very important information. In 60% of angiosperms, pollen grains are shed in two-celled stage. So in 60% of angiosperms, pollen grains are released in two-celled stage, where there are only something called as vegetative and generative. In remaining 40%, it is three-celled stage. Three-celled stage. And students, remember, this entire pollen grains can be stored in liquid nitrogen at minus 196 degrees. Okay. Now, students, how many of you, as soon as the dust comes, you're like, achi, achi, achi. How many of you start? Okay, students, quickly. How many of you start sneezing as soon as you come in contact with the dust? If you like me, you have dust allergy. But there are people who have something called as pollen allergy. As soon as the pollen grains travel in the air, and by mistake also, if the pollen grain goes inside their nose, gone. They'll start sneezing at the end of time. They'll keep on sneezing. Whole life they'll keep sneezing. Yeah. So that is very, very important. That is called as pollen allergy. There are certain plants which can release pollens which can cause something called pollen. But students, do you think pollen grains has advantage also? Can you eat pollen grains? There is something called as pollen tablets. Pollen tablets which are very famous in your England and European countries, where people take it as athletes take it, and also it is given to horses. Okay, horses. Pollen energy. Now there is something called as pollen viability. What is the meaning of pollen viability? Now imagine the pollen grain has been released. How long can it remain active? And you know, as soon as it comes in contact with the stigma, it can produce pollen. That is called viability. It remains active. Okay. Now, for some cases, it is very short duration. In some cases, it's very large duration. For example, understand. Pollen grains can remain viable for various duration. For cereals, it is around 30 minutes. And for members of Solanaceae and Rosaceae, for almost. Now, can anyone tell me, students, sir? Can anyone tell me one family is missing here? Which is that family in the? Okay, deliberately, I am not. I don't understand, students. Now we come to the entire gynoecium. The gynoecium is made up of pistil and a carpel, right? The carpel here has three main parts: stigma, style, and ovary. Stigma, style, and ovary. Here, diagram. One carpel has something called as stigma, style, and now please do not get confused right now. Sir, what is this structure here? Understand, I'll tell you. Leguminaceae. Okay. Now understand, students. What exactly is this structure and where is this structure present? I'll show you a small diagram and this entire doubt will get clear. All of you just follow me. Okay, students, I'm not able to turn. Do you realize? Because if I turn a lot, the voice will start to break. So I'm become a robot today. Robotic I've become. Okay. Hopefully it doesn't break now. Now students, all of you know this is the stigma, style, and the ovary. All of you just listen to me. This is your style, right? Now listen to me, all of you. Inside this ovary, there can be many ovules. Can all of you see the ovules now? These structures on the inside, these structures are called as the ovule. And can all of you see this entire ovule right here? It has a stalk right here. Hope all of you can notice this region right here. This is called as the stalk of the ovule. Stalk of the ovule. Okay, stalk of the ovule. Yes, stalk of the ovule. Okay. Now this structure right here, try to imagine that structure. The stalk here. This entire stalk is arranged this way. This is called the stalk of the ovule, and the stalk of the ovule, this anatropous ovule here is called as your tunicle. Tunicle is the stalk. Tunicle is the stalk, right? Tunicle is the stalk. Yes, we will cover all chapters. Don't worry. Stay till the very end. Like the video first of all of you. Stay till the very end. We will do it. If students are not there, then we will not do it. Simple as that. Now, this is called a stalk. Then, now this entire stalk is attached to the ovule. The point of attachment is right here, and the point of attachment with the stalk is attaching with the ovule. That is called as your hilum. Point of attachment. Okay? Now understand all of you. This entire ovule right here has something called as outer protection and inner protection. The protection here is called as integument. Outer integument, inner integument. Now can anyone tell me in the chat, students? Do you find these integuments in your pteridophytes? Why? Because if integuments were present in pteridophytes, had to produce. And these integuments are very important because they form the seed coat eventually. Seed coat eventually. Okay. Now listen to me all. Now these integuments do not completely combine or join together. There is a small region here where integuments do not join, and this region is called as micropyle region. The region completely opposite to micropyle is called as chalazal region. Now here there are multiple diploid cells throughout diploid cells. They are called as nucellus, and right in the center we have the embryo sac. Right in the center we have the embryo sac, and embryo sac right here, right in the middle, that is called as female gametophyte. Your ovary is not the female gametophyte. Ovule is not the female gametophyte. It is the embryo sac which is the female gametophyte. Okay. Yes, students. Now, if you look at this diagram right here, students, you will notice that certain types of, right, in certain cases, the carpels can be free and the carpels can be fused. When the carpels are free, it is called as apocarpus. When the carpels are fused, it is called as syncarpus. Imagine carpels as your fingers. If I join all the fingers, it becomes syncarpus. If I make it free, it's called as apocarpus. Important diagram. This is very important diagram for all of you. Very important. Here, understand, students. Understand examples. You understand. Here. Here. Multicarpal is syncarpus. Here. Syncarpus. Example is papaver. Papaver is going to be syncarpus, and this is an example of your apocarpus here. Apocarpus. Okay, mycelia. Mycelia is going to be your apocarpus, syncarpus. Integument is done, nucellus done, done. Embryo sac is megaspore, done. Embryo sac formed from a megaspore, done. Now, students, we will start with your megasporogenesis. What is megasporogenesis? Megasporogenesis is a process in which your entire male gametophyte is formed or the embryo sac is formed. Now, if you remember, students, can anyone remind me one more time? The entire microsporogenesis, where does it start from? Anyone remind me? Where does the microsporogenesis start from? Now, if you look at the megasporogenesis, megasporogenesis starts from a diploid cell called as nucellus. The nucellus it starts. Okay. From the nucellus. Now, what happens here? One cell from the micropylar region becomes enlarged, and that is called as MMC. What is MMC? MMC is nothing but megaspore. Now, this megaspore mother cell is diploid, undergoes meiosis 1, meiosis 2, and finally give rise to 1, 2, 4 haploid gametes called as megaspore. Now, there is something called as selection here. What is selection? Out of these 4 megaspores, 3 will completely degenerate. Only 1 will be functional, and this type of selection, where out of 4, 3 is degenerating, 1 is succeeding, that is called as monosporic development, and this is called as functional megaspore. Now, understand, students, this functional megaspore now undergoes multiple. For example, this functional megaspore now undergoes successive mitosis, one by one mitosis. So, first we have mitosis 1, where it's completely free nuclear. What do I mean free nuclear? The meaning of free nuclear is that there is no cytokinesis here. There is no cell wall formation happening here. No cell wall formation. Only nucleus is dividing here. Okay, you can see nucleus dividing. One nucleus, two nucleus. So, two nucleated condition. First mitosis. Second mitosis, 4. 2 to 4, 4 to 8. 8 to 16. Okay. 3, 4, 5, 6, 7, 8. 8 only. 1, 2, 3, 4, 5, 6, 7, 8. 8 is the maximum here. Okay. So, 8 nuclear condition. Now, this 8 nuclear condition undergoes polarization. What do you mean by polarization? Polarization means it is going separation. Then we have your cell wall formation. After cell wall formation, we get all cell walls everywhere here. And finally, we have the entire male. Now, students, again, in this female gametophyte, if you notice, this is a very, very important diagram. Not even turn, this is the maximum I can. It's a very important diagram for all of you. Right? Mike. In this diagram, if you can notice, students, they can ask you the ploidy level here. They can ask you the ploidy level. If they ask you, students, what is the ploidy of antipodals, can anyone tell me what is the ploidy of antipodal cells? Antipodal cells here are completely haploid in nature. That those are present towards chalazal region. Now you have to even remember this. What is present towards chalaza? What is present towards micropylar? How do I remember it? Okay. Now it is very summer. It's very summer season now. Read AC. A, C. A for your antipodal, C for chalaza. AC. Remember it as that. AC here. Now, at the complete opposite end, that is called as micropylar region, we have 3 main cells here. One is called as a synergids, two synergids here, and one egg cell. This synergids and egg cell together they are called as egg apparatus. Here every single one of them is haploid in nature. Now again students, in the synergids students, we have something called as filiform apparatus here. All of you see here, this is called as filiform apparatus. Now, some of you might ask me, sir, what is the function of filiform apparatus? The main function of filiform apparatus is that it helps in guiding the pollen tube. The pollen tube has to go inside. So it helps in guiding the pollen tube. That is your filiform apparatus, right? Yes, students? Now, right in the center, we have two nuclei. They are called as polar nuclei. So these two polar nuclei, students, these two polar nuclei will completely, and when these two polar nuclei, when they completely fuse together, that is called as your central. Now, some of you might ask, sir, what is the ploidy here? The ploidy of embryo sac. The ploidy of embryo sac is haploid, but the only diploid part here is going to be the central cell, which is diploid in nature. All of you can just follow through this also. Now, so this is one famous question here, which comes as 2 mark also. Sir, how many cells and how many nucleus do we have here? In this entire cell, how many cell and how many nucleus? Let's count. How many cells first? 1, 2, 4, 5, 6, 7. There are 7 cells here. But how many nucleus, students? 1, 2, 3, 4, 5, 6, 7, 8. So there are totally 8 nucleated, 7 celled, 8 nuclear condition. Everything is given, you can just go through this. This is summary of everything. Okay. Next thing is called as pollination. Pollination is mainly of 3 types. Okay. Autogamy, monogamy, and gametogamy. Understand the difference first of all. Okay. What is autogamy? Let's not take cross pollination. Let's say just take pollination first. Okay. What is autogamy? For example, if this is a flower, if pollen grains are traveling from here to the same flower, that is called as autogamy. Transfer of pollen grains from the anther to the stigma of the same flower, autogamy. What is gametogamy? Understand the difference, gametogamy. If both of these flowers are present on same plant, if they are present on the same plant, if both of them are present in the same plant, from here to here if they go, that is called as gametogamy. Then what is xenogamy then? What is xenogamy? Understand. Xenogamy is nothing but if both of these flowers are present on different plants, that is called as gametogamy. From here to here, different plant, that is called as gametogamy. Okay. Now we have something called as chasmogamous flowers and cleistogamous flowers. Difference you understand? They can ask you in 1 mark question. What is going to be chasmogamous are normal flowers which open very brightly. What is cleistogamous flower? Understand this term. Cleistogamous flowers are the flowers which do not open. These flowers are present underground. And listen to me very carefully. Cleistogamous flowers are the flowers where we only have autogamy. Why? Because if the flower doesn't open at all, then there's a problem. Doesn't open at all. So there is only self-pollination only. Only self-pollination. Okay, only self-pollination. Closed flower. We can also call them closed flower. Now here they can ask you examples. Example is going to be your Viola. It's given Viola. It's given Viola oxalis communalia. Key examples of the cleistogamous flower. Okay. Examples of pollination they will not ask you. Don't worry. Okay. Now can I tell you one more thing, students? Is it a disadvantage also, this flower? Do you think it's a disadvantage or it only has advantage? Do you think this autogamy, repeated autogamy, does it have a disadvantage? Yes. Advantage is that always pollination will happen. Assured set of seeds, always. But does it have a disadvantage? There is something called as inbreeding depression. What is inbreeding depression? If there is a continuous self-pollination, continuous self-pollination, there could be something called as accumulation of recessive alleles. Recessive alleles can happen. If that accumulation happens, right, that can lead to inbreeding depression. As advantage and disadvantage also. Yes. Advantage and disadvantage both is there. Now, imagine there is a flower. There is always self-pollination. Now we have to stop it. Instead of doing inbreeding, we have to do outbreeding. Now there is something called as outbreeding devices. What is that? These are different methods to make sure a flower does cross-pollination. Now what are those? Understand. The first one is called as, there are four main things here, understand, okay, four main things. How many? Four main things. The first one is called as heterostyly. What is heterostyly? Very simple to understand. What is heterostyly? Understand. Heterostyly. In heterostyly, what happens? If this is the entire pistil, your anther, your entire stamen is somewhere here. This right here. Will the pollination happen here, students? Will the self-pollination happen? Not happen. Self-pollination will not happen. It can very rarely it can travel up here, but it's very rare. It does not happen at all. Okay. Very rarely it happens. The first one is called as heterostyly, where the stigma, where the entire style is very long and anther is very short. It can be other way around also. Where anther is very tall and your gynoecium is very or your pistil is very small. Other way is also possible. The second one is called as maturity. What is maturity? Sometimes what happens is the stigma is ready. Stigma is very receptive. I'm like, I am ready. But pollen grain is not released. Pollen grain is not released at all. Okay. The girl is telling you, I am so ready to get married. But the guy is like, no, I can't get ready. Not possible. That is called as your, or the other case also, when the guy is ready, like, I am ready now. I want to get married properly. A full grand wedding. But the stigma is like, I am very young. Stigma is like, I am very young, I am not ready. Okay. Don't worry about technicalities here. Remember the concept here. In one done. The third one is called as self-incompatibility. What is that? Self-incompatibility is a much more genetic one. Okay. Now, last one is going to be production of unisexual flowers. Self-incompatibility, stigma at different position, absence of synchrony, that is maturity period. So all of these four things which you need to know. Unisexual flowers, heterostyly, not say that I get confused. Maturity one and the self-incompatibility. Okay, four things which you. There is a famous 3-mark question. I will show you exactly now what you need to write. Okay. All of you know, students, there is something called as agents of pollination. What is that? There could be wind, there could be water, there could be insects. Right. Now here, water is not very important. Right. Water, all you need to remember water is that the example of Vallisneria. But when it comes to wind and insect, okay, there is something called as characteristics of wind pollinated and characteristics of insect pollinated flowers. That you need to remember, and I'll make a I've given a slide for you. Just remember that and go. Okay, see this now. Before we go to wind and insect, let's finish water. Pollination by water is quite rare. It's quite rare in flowering plants and limited to about 30 genera, mostly monocots. So mostly monocots it happens. Okay. Now, first about what about Vallisneria? How does it happen in Vallisneria? Understand. How does it happen in Vallisneria? Now, this is your Vallisneria concept here. You can notice, students, all of you can notice very clearly here that the entire male flower is present underwater. The female flower is present on the top. Female flower on the top, male flower underwater. Why? Female flower is on the top because of a long stalk. Long stalk, it is not. So what happens? Here, the male flower will produce the pollen grains. The pollen grains will have a mucilaginous sheet around it. Like, waterproof liquid is there. It has a waterproof covering. Okay. Eventually, all the male gametophyte or pollen grains go on top, and there will be fertilization. This happens in the valsinate. In the case of Zostera, it is completely underwater. Students, I feel all of you listening to one podcast, no? Continuously, I'm talking from last almost 45 minutes. I feel all of you listening to one podcast. All of you are sitting and listening to what I'm saying. Students, anytime you have any doubt, put in the comment section. I will take it. It is not recorded, it is live. Okay. Now there is two more questions which they can ask you. Two more questions. In most of the water pollinated species, pollen grains are provided by a wetting form called as mucilaginous covering or mucilaginous sheet. Okay, just remember this mucilaginous sheet present in your water pollinated. Okay, but there are some special cases. What are special cases? These flowers are present in water, but they don't want to do water pollination. I don't know why. Okay, so aquatic plants as water lily and water hyacinth are insect pollinated. Okay, now students, I'll tell you now what are characters. Understand the characters now. Characters you understand. In my class, listening to a podcast. This entire slide right here is showing you all the characters of your wind pollinated flowers. Small, inconspicuous, colorless, odorless, nectarless flowers. Flowers are packed into inflorescence. Pollen grains are light and dry. Pollen grains are dusky and non-sticky. Well exposed stamen, feathery stigma, uni-ovulated pistil. Means single ovule in the ovary. So all of you remember this. Take a screenshot. If you want the notes, I will send all my notes on this telegram right here, students. My telegram is right here. In this telegram, I will send all the notes. So download it today. Revise once before going to exam. Okay. These are the characters of your insect pollinated flowers. Can all of you see? Showy and bright colored flower. Small flower grouped into conspicuousness. Landing platform. Nectar guides. Produce odor or foul smell. Foul smell in the case of your, Amorphophilus, right? All these large flowers can also produce foul smell to attract the flies. Okay. Secretion of nectar, production of edible pollen. So this entire thing is also called as reward mechanism. You are literally bribing the entire your insects to come and do your work. You are paying them actually. Not bribing, you are paying them. Okay. In some species, floral reward are providing the safe space to lay eggs. Example of that of tallest flower called as Amorphophilus. Here, reward mechanism is a space for laying the eggs. You might have seen, right? Sometimes what happens is when a lot of people are coming to work for you, you don't even just pay them, you also give them a house to stay. Someone is coming to work for you, sometimes you give them a house to stay also. In government jobs, they give you a house, quarters. Like quarters, you might have been quarters, right? They give you quarters. In army also they give you quarters. Similarly, the work is being done, they're giving you a house to stay also here. Okay. In depression, I told you already. Now, student, there is something called as pollen-pistil interaction. There is something called as pollen-pistil interaction. Understand what is pollen-pistil interaction? Understand, very simple. Many students usually skip this part because they think it's okay. Now, I'll ask you one question, all of you. Right. On a mango flower, right? If a pollen grain of rose comes and fall, will the pollination happen or not? Tell me. Examples of mutualism, students. Mutualism example is very simple. You can take your moth and Protumbus moth and fig fruit, and that is there. The yucca plant, yucca and the moth. There is wasp and fig also. You can write wasp and fig also from your biodiversity, your organism population. Okay. What happens, students? Tell me. Can a mango flower be pollinated by rose pollen grain? Is it possible? Why? Because students, there are some receptors. Every pollen grain and stigma has something called as chemical dialogues. So when the pollen grain lands on the stigma, it exchanges some dialogue exchanges there. Some dialogue exchange. Dialogue exchange is there. Now what happens is when this dialogue exchange is happening, it will confirm. Like same species, entry is there. Different species, go away from here. Go away from here. That is called as chemical dialogue exchange, and that will determine something called as compatibility or incompatibility. Okay. Pistil has the ability to recognize the pollen whether it is compatible or incompatible. Pollen grains germinate on the stigma to produce pollen tube to one of the germ pore. So from the germ pore, what happens? The pollen tube is coming out. Okay. The pollen tube, after reaching the ovary, enters the ovule through micropylar and enters one of the synergies through filiform apparatus. Okay. Now, all the events from the pollen deposition, all the way from the pollen deposition, right? Pollen deposition, right? All the way from the pollen deposition, starting when the pollen grain is coming and landing, from that point, from the stigma until pollen tube entry, the pollen tube enter the ovule are together referred to as pollen-pistil interaction. That's all. When the pollen tube is landing on stigma, all the dialogue exchange, pollen tube growth, and the entry of pollen tube inside the ovary, inside the ovule, through inside the embryo sac, through synergid, entire event is called as pollen-pistil interaction. That's all. That's all, students. Okay, the entire thing is called pollen-pistil interaction. I will show you the diagram for it. Can all of you see this diagram, students? The pollen tube landing here, pollen tube growth, entry of pollen tube into the embryo sac through a synergids. Degenerating synergid is entering. Okay, entire thing is called as pollen-pistil interaction. I'm telling you. There is something called as artificial hybridization. Why do we need to do artificial hybridization? First, well, understand. How many of you like mangoes? Raise your hands. How many of you like mangoes? Students. How many of you like mangoes? So I love mango. I love mango. Now, what happens in nature is that, let's say I have one mango. It's very big, but not sweet at all. Not sweet at all. I have one more mango. It's very sweet, but it is this much only. So what do is I do something called as crossing of these two. So when I do a crossing of these two, there is a chance, not 100%. There is a chance that the mango which we get will be very large and it will also be very sweet. Best of both the things into one. Okay, you will get a large mango also, sweet mango also. That is the reason we do artificial hybridization. How do we do it? For example, first of all, we have a bisexual flower. And this flower has both the stigma, style and ovary, and also it has the entire stamen. Now students, if I take a bisexual flower, is there a chance of self-pollination? Tell me in the chat. Is there a chance of self-pollination? If I take a bisexual flower, is there a chance of self-pollination? Like, yes sir. So what do we do? Chop chop. What do? We completely remove all the entire, the anther is removed. The anther is removed because anther has the pollen. So all the anthers are plucked out, making the flower unisexual. From bisexual to unisexual, you made it off. Removal of anther. That step is called as emasculation. Emasculation. It's given here. Emasculation. Now students, after emasculation is done, what do? You need to cover it. That is called as bagging. Why? To make sure that some random pollen grain will not come and land there. That is called as bagging. After bagging, we have something called as tagging. What is tagging? In that we add a tag there, like a name card, identity card to it. Name, species, which pollen grain, date of pollination. Okay. Then we add our desirable pollen grain and we do rebagging. That is called as artificial hybridization. All of you can write this in your own words. You will get points. Get marks. Sorry. Okay. The thing is, students, there is always a chance in normal traditional hybridization that you will get something called as your undesirable traits also coming in. Can anyone tell me which method can be implemented to make sure we only get desirable products? Anyone, students? Can anyone tell me, is there a chance that we only get desirable product? We don't want undesirable thing only. Is there a chance? And that you will learn in biotechnology chapter. Biotechnology. Yeah, biotech you will learn. During the artificial hybridization, after entering one of the synergids, the pollen tube releases two male gametes. So once the pollen tube is here, after entering here, all of you see here, it will release two male gametes here. Release how much? It will release two male gametes. Two male gametes. Now understand all of you what exactly happens here. Okay. Now in this pollen tube is entering, filiform apparatus synergids. Two male gametes are released. Now one male gamete here. The first male gamete will fuse with the until your name. Okay. So students, you tell me in the chat. In unisexual flowers, do we need to do emasculation? Tell me yes or no. In unisexual flowers, do we need emasculation? No. Emasculation not required. Unisexual flowers not required at all. Okay. Now what exactly happening here? One of the male gamete is fusing with the egg. One male gamete plus egg, that will result in the zygote formation. And this step is called as syngamy. First type of fertilization happening here. Here, first type of fertilization is happening. The second male gamete. The second male gamete will travel up. The second male gamete will fuse with the central cell. Okay, second male gamete, two polar nuclei called a central cell, which is diploid in nature, and we get PN. Here N plus N is 2N, N plus 2N is equal to 3N in nature. And this PN will develop into P's, primary endosperm cell, and eventually the endosperm. And remember students, remember always, this zygote will develop into embryo later on. So there is a small concept here. The embryo will never form, generally never form until endosperm is developed first. Embryo will always wait for the endosperm to form first. Why? Because endosperm can provide nutrition to the growing embryo. That's why embryo will wait. Embryo will wait always. Okay. Now students, this entire process is called as triple fusion. Why triple fusion? Because 2N here, N here. So triploid tissue, that's why it's 3N. So triple fusion. Syngamy. Together it is called as double fertilization. Together it's called as what? Double fertilization. So if they ask you a question examination, explain double fertilization for 5 marks. For 5 marks you need to draw this diagram, write this entire thing and get the 5 marks. And all of you, students, can I get some fire emoji in the chat if you're understanding? Because I'm talking constantly from last 1 hour. From last 1 hour I'm talking continuously without taking a break. All of you. If you're understanding, some quickly fire emoji in the chat. Yes, all of you. See, this is called as a revision session. I'm doing revision. All of you might know the concept by now. It is proper revision technique. Fast track revision. Okay. Done. Now here a small concept applies here. The coconut water, what exactly happens in coconut water? Understand. In coconut is there, all of you know coconut? The drupe. Right. In coconut, what is happening here is that endosperm is very special because there are two types of endosperm in coconut. One endosperm is called as cellular, one is called as free nuclear. In coconut, when we take a coconut, students, we drink this water out of it, water, right? The water or the liquid part is called as free nuclear endosperm. Okay. Water coconut is free nuclear endosperm, and the surrounding white part is called as cellular endosperm. The white part which you eat, right, that is called as cellular. The liquid is called as free nuclear. Please remember this. Please remember this. What is the difference? The difference is very simple. In free nuclear, the nucleus will keep on dividing, but there will be no crosswell formation. We call that something called as syncytium formation. Syncytium formation. Okay, no cytokinesis. Basically, no cytokinesis, only chirokinesis. In free nuclear, continuous chirokinesis happens. Friends, I hope I'm not scaring you with difficult words. Okay. Now, embryo development at the micropallar end of the embryo sac where the zygote is situated. The portion of embryonal axis. If you look at the developing embryo will look like this. It has something called as the root development and shoot development. Near the shoot we have something called as epicotyle. Above the root we have the hypocotyle. And these two structures are called as cotyledons. Now can anyone tell me in the chat, is this the structure of your monocot or dicot? Anyone? Anyone students? Anyone quickly in the chat? Structure of monocot or dicot? This is the structure of your dicot. Why dicot, students? There are two cotyledons here. Two cotyledons is a dicot. Cotyledons. In grass family, a single cotyledon is called a scutellum. So in the case of monocots, there is a single cotyledon is present. In the case of monocots, we can see a single cotyledon. All of you see this? What is good? Monocot. All of you see this, students? This diagram right here. This one is a monocot. All of you see here? Monocot one. Why monocot, students? Here we can see a single shield shaped cotyledon called as scutellum. Scutellum is a single cotyledon in the case of monocot here. Now here, this entire thing is called as endosperm. Here we have an aileron layer called as protein layer called as aileron layer, and here radicle is covered by a protected by a layer called as coleorhiza. Plumule is protected by something called as coleoptile. So if you find it difficult to remember who's protecting who, very simple hack is there. Coleorhiza, R is there, radical, R. Coleoptile, P is here, and P for plumule, PP. So very easy to remember. Coleoptile, plumule, coleorhiza, radical, RR. Understood all of you? Here you can see dicot seed. Here you can see dicot seed also. Dicot seed. Two cotyledons. Can you see two cotyledons? Two cotyledons is present. Here. Now, all of you remember nucellus? Can anyone tell me, students, what was nucellus? What is the ploidy of nucellus? The ploidy of nucellus. Anyone ploidy of nucellus? The ploidy of nucellus was diploid, right? In some cells, right, in some of the organisms, right, nucellus does not perish. Nucellus becomes persistent. When the nucellus becomes persistent, that is called as perisperm. So in some seeds as a black pepper and beet, residual persistent nucellus is called as perisperm. So remember, okay. As a seed matures, it becomes relatively dry and contains 10 to 15% moisture by mass. The wall of the ovary develops into wall of the fruit called as pericarp. So this basic thing all of you need to remember, right? Ovule develop into seed. Ovule will develop into seed, and ovary will develop into. Now this ovary has a wall, outer wall. This outer wall will develop into something called as your pericarp. Develop into pericarp. Okay. Now there is something called true fruit and false fruit. What is true fruit? True fruit is a fruit which develops from the ovary only. The ovary only. What is a false fruit? False fruit is a fruit which develops from the ovary plus other part such as thalamus. The example is apple, strawberry, cashews. Right? Apple, cashew are called where the thalamus can contribute to fruit formation called as false fruit. So there is something called oldest viable seed, that is lupinous articles, that is actually from the Arctic. Old seeds, 2,000-year-old viable seed again in Phoenix Lactifera was discovered by King Harold Palace. This much they will not ask you generally. There are two more concepts which they can keep on asking you. Right. Very simple. PEN is primary endosperm cell. Primary endosperm nucleus. First there is nucleus formation, eventually it becomes cell completely. Okay. Now understand there is something called as concept called as apomixis and there is something called as parthenocarpy. What is parthenocarpy first of all? Parthenocarpy is formation of fruit without fertilization. Parthenocarpy. What is apomixis? Apomixis is formation of seed without fertilization. Given right here. Okay. Apomixis is the asexual reproduction that mimics sexual reproduction. Development of seed without fertilization happens here. Okay. What is parthenocarpy? Formation of fruit without fertilization. And again remember students, in parthenocarpy you will always have seedless fruit. Okay. Now there is something called as polyembryony also. Polyembryony. What is polyembryony? Polyembryony is, poly means many, embryo. Many embryos are there. Polyembryony. Okay. Example is going to be citrus. There will be many, more than one embryo. Okay. And students, with that, entire chapter fast track revision is over for your sexual reproduction of flowering plants. I want one word. How was it? Parthenocarpy examples you can write watermelon, banana, and everything. Polyembryony is going to be done. That's all. In just 1 hour, almost 1 hour, we finished entire thing. Yes, oxygen is involved. Yes, for plant growth related. How is it, students? Full chapter, crazy revision we did. Now, can we start with human reproduction, students? I will finish human reproduction in the next 40 minutes. Can we start? Can we start now? Human reproduction. All of you. Can we start, all of you? Friends, all of you know. See, now you are learning the concept. Now you are learning the concept, right? Exactly at 6 p.m. today. Exactly 6 p.m. today. What do you have, students? Exact 6 p.m. What do you have 6 p.m. today? Exactly 6 p.m. today. Voice breaking again? Hold this now. So exactly 6 p.m. today we have question practice. So all of you will attend that, because see, now you will understand the concept. But if you do not know how to write the answer, how much to write the answer, what to write, you will not get marks. So evening class, question practice, morning class, concept. Okay. Human reproduction, very easy, students. My favorite chapter, very simple chapter. Okay. Now, I want everyone just focus the next 40 minutes very carefully. Listen to what I am saying and you will understand the whole chapter. 40 minutes. Give me 40 minutes. Just 40 minutes, entire thing I will finish. Can we start, all of you? Can we start? All of you? Okay. In the case of human reproduction, okay, there is something called as a male reproductive system and female reproductive system. All of you need to remember, students, in the case of humans, there is something called as sexual reproduction. In sexual reproduction, there is always involvement of, there is something called as gametes. When the gametes fuse, it results in the formation of your zygote. Now, in the male reproductive system, the female reproductive system, both are located in the pelvic region between the two coxal bones, present in the pelvic region. Okay, pelvic region. Now, here in the case of male reproductive system, right, it has something called as primary sex organ and secondary sex organ. Can anyone tell me which is the primary sex organ here? The primary sex organ in the case of your male reproductive system is nothing. It's nothing but the testis. Primary sex organ. Primary sex organ. Now, students, every primary sex organ has two main roles. It should be able to produce hormone, should be able to produce gametes. Testis here is able to produce the gametes, that is a sperm. It can also produce the hormone, that is androgen, testosterone. This entire testis is located in a pouch like structure called a scrotum. You might ask me, why sir, why is the need? because the formation of sperm called spermatogenesis requires 2 to 3 degrees less than the body temperature, and that is provided in the scrotum. Here, inside each testis, students, inside each testis, there are 250 compartments. Like imagine there's a big house. In that one big house, there are 250 rooms. 250 rooms in one big house. The 250 compartments are called as testicular lobule. Called as what? Testicular lobule. Now, one big house. In that one big house, there is something called as your 250 rooms, and inside every room there is 1 to 3 window, one testis. Inside each testis there are 250 compartments called as testicular lobules. Inside every testicular lobule there is something called as 1 to 3 highly coiled structure called as seminiferous tubule. Now inside the seminiferous tubule there are 2 cells. Inside seminiferous tubule, students, there are 2 cells. Yes, students, yes, not all chapters will do high weightage. Ecology, all of you can study, right? Ecology, I don't think it required. Ecology, I'll show you important questions today. Evening, ecology, we won't be teaching entire ecology. Ecology, we will give you questions directly. Okay, we will teach you high weightage because all of you know, right? CBSE, no. Okay, all of you know the main weightage comes from these 2 chapters and genetics. We'll mainly focus on high weightage topics, high weightage chapters. Okay. All of you listen to me. Inside seminiferous tubule, there are 2 types of cells. One is called as male germ cell, also called as spermatogonia, and the second cell is called as Sertoli cell. Okay, Sertoli cell as well as male germ cell. So you might ask me what is the function. This male germ cell is diploid in nature. Will undergo spermatogenesis to produce the sperm, and the Sertoli cell will provide nutrition. Okay, and it also helps in spermiogenesis. Okay. The male axillary ducts include radial testis. There are multiple ducts here. I'll show you the diagram, students. Okay. All of you focus now. Very important diagram. Very important diagram. So if you notice here, students, this is the male reproductive structure. Here we can see the primary sex organ, that is the testis here. Testis is able to produce the sperm. The sperm is taken up by the first place it goes is radial testis. From here it goes to vasa efferentia, epididymis, vas deferens. The sperm is traveling up. Now eventually the sperm reaches a region right here called as ejaculatory duct. Ejaculatory duct. Now in this ejaculatory duct right here, one more duct is coming from top, and this duct is coming from the seminal vesicle, which is the first gland here. From here secretion is coming from here also. So this middle point is called as ejaculatory duct. And students, remember students, seminal vesicle produces certain secretions. It is rich in fructose, rich in certain type of prostaglandins, and also it makes up the 60 to 70% the semen. Okay. And although it is present as pair. Then we come to the second gland, that is called as prostate gland. It mainly functions as alkaline fluid, produces alkaline fluid, and also it produces certain types of enzymes. But prostate is going to be present as single. Then we have bulbourethral gland right here. Bulbourethral gland mainly produces certain types of mucilage, helps in reproduction, during reproduction. Okay. 3 glands and 3 glands, all the ducts here. Finally, from here the semen is traveling. See here, why do I call it semen here? The reason I am calling it semen because it has something called as seminal plasma. Seminal plasma is basically made up of secretion of the seminal vesicle, prostate and bulbourethral. All 3 is packed in the seminal plasma. Thus the seminal testis. Together it is called as So from here the entire semen is being travelled through the common duct called as urethra. is a common death for release of urine and the semen. Finally, it is coming out of the opening called as urethral meatus. Urethral meatus, that is the opening here, and this entire swollen end of the penis is called as glans penis. The entire penis is covered by a loose skin called as foreskin. Try drawing it in once and then learning it once. It's fine, it will not take much of time. Okay. Now we come to the friends, you will get this notes, don't worry. Male axillary glands done, so male vesicle. Now we come to the female reproductive structure. The female reproductive structure mainly consists of primary sex organ, secondary. The primary sex organ here is the primary sex organs are the ovary. The primary sex organ are the ovary. Now imagine every month one ovary will release one egg. And alternate, if for example, if the one January this ovary is releasing egg, February this ovary will release egg. They will alternate among each other. Okay. Now all of you understand here, students. So if the egg is released here, the entire egg is picked up by a small hole ostia here with the help of a finger-like structure called as fimbriae. The fimbriae will help in picking up the egg. Now once the egg is picked up from here, the egg will travel to a much more cone-shaped structure called as infundibulum. Then from here it is travel up, that is called as ampulla region. And here, this is the same region where the fertilization will occur. Now from here it will travel to a much more narrow region called as isthmus. And this entire three regions are part of fallopian tube, also called as oviduct. Second user, yes, exactly, second user. Now from here the entire, if zygote will move, if egg will move. Okay. Now egg or zygote is moving here. If the egg is not fertilized, it will be removed as such. If there is fertilization, the zygote will actually attach at this point here. That is called as implantation. Now understand, students, this middle region right here is called as uterus. It is called as uterus. And this uterus right here, students, has something called as three layers here. It has something called as outermost layer called as perimetrium, middle layer called as myometrium, innermost layer called as endometrium. What is the difference? Understand. Perimetrium is very thin. Myometrium is made up of smooth muscle cell and it undergoes contraction during childbirth. Okay. While your endometrium is very secretory in nature, it is glandular and it undergoes changes during every single menstrual cycle. Then there is a small opening here, is called as cervix, and cervix will open into vagina through cervical canal here. So the cervical canal plus vagina together they form the birth canal. Okay. Done. Shape of the uterus is inverted pair, remember that. Birth canal, done. 3 layers of tissue, endometrium, done. There is something called as external genitalia also. The external genitalia is called as the vulva. The vulva has multiple parts. For example, the outermost part is called as mons pubis, made up of female external genitalia, which is a cushion of fatty tissues covered by skin and hair. There is the outermost layer. Then labia majora, labia minora. Labia majora are fleshy foldings of tissue, then down from the mons pubis and surrounding the vaginal opening. Right. Labia minora are paid foldings of tissue under the labia majora. So there is something called as labia majora, and under it there is labia minora. Okay. Now opening of vagina is often covered by a partial membrane called as hilum. Again, students, Phylum, presence and absence of phylum doesn't determine the virginity, because there are many cases where the entire hymen is broken because of other activities such as your horseback riding or play. So it is not. Okay. At the end of the female reproductive system, right, or the genitalia, at the point there is something called as a tiny finger-like structure called as clitoris. Okay. Upper junction of two labia minora, clitoris is there. Okay. Above urinary opening. Now here also, students, remember in the clitoris also, the clitoris there is something called as erectile tissues are also present. Erectile tissues are also present. Here. Now, one more thing which they can ask you question here, students. They can ask you the ducts, fallopian tube here. They can ask you the endometrium layers, and that's it. Nothing more. Okay. Now, next thing which I have to add, one more thing I want to tell you, students, what I was speaking. I remembered, forgot in my head right now. Never mind. I remember, I'll tell you again. Okay. Next, one of the secondary sex organs here is called as the breast or the mammary glands. Right, the breast or the mammary glands. You can see this is of your breast or the mammary glands. Entire breast or the mammary glands is made up of multiple cushion or tissues. It is made up of a lot of fat also. Okay. The glandular tissue of each breast divided into 15-20 mammary lobes. There are multiple 15-20 mammary lobes. All of you can see this entire mammary lobe. Mammary lobe. Entire thing is called as mammary. Mammary lobes are present, containing clusters of cells called as alveoli. Now they can ask you a question. There is milk produced. The milk is produced in the alveoli and it is stored in the cavity of alveoli itself. There is a small point you have to remember. Okay. In mammary glands, mammary tubules of each mammary lobe join to form mammary duct. Called as, first we have mammary lobe. Mammary lobe is there. Mammary tubule, mammary duct, mammary ampulla, lactiferous duct, okay. And finally the milk is coming out of, then we have where the milk is coming. This is a small flowchart here. Mammary lobes, mammary tubules, mammary duct, mammary ampulla, lactiferous duct, and nipples. It is small way where the movement of milk is. You can ask this. Okay. So, ma'am. Hi, ma'am. Oh, you did this.
Hi. Is it going? Yeah.
Entire chapter, right? Realize we have taught this entire unit for 6 hours, I believe, right?
Yeah.
So if I'm trying to do the entire 6 hours chapter in 1 hour or 2 hours, it takes a lot of skill and effort, ma'am.
So how am I going to do molecular basis?
Ma'am, I'm talking about any break I took. Past
Okay.
1.5 hours, I'm talking. Right.
I just finished the crash. Once crash, I'll come schedule. Okay. But biotechnology chapter I'll take. Okay.
Ma'am,
Yeah. Taking biotechnology. Don't. One of us will take. When the session will get over, you come at the end of the session.
okay, ma'am. Bye, ma'am. Gone already. Okay, students. Now, all of you need to be very careful when you're trying to learn the spermatogenesis. It's very, very important. And I'll teach you in a very brief way follow through and you will learn it. Okay. Ma'am voice is breaking because ma'am was not holding. Students, you realize from that time I'm just holding this, I'm teaching, right? I have to tell this to ma'am also. Now, students understand. Spermatogenesis. Where does it start from? The entire spermatogenesis starts at the embryonic, starts at the puberty. Entire spermatogenesis starts at the puberty. Initially, we have a cell called as your male germ cell, also called as spermatogonia. Can anyone remind me, students? Can anyone remind me? Male germ cell, also called as spermatogonia. Where is it present? Anyone who can remind me? Where is it present? Is it present inside seminiferous tubule? Yes or no? Is it present inside seminiferous tubule? Yes or no, students? Quickly in the chat. It is present in the seminiferous tubule, right? One of the male germ cell or spermatogonia undergoes mitosis, increase in number. Now, one such male germ cell will grow in size, and we call it primary spermatocyte. Now, this primary spermatocyte undergoes meiosis to produce two cells. And we call these two cells, which are haploid in nature, secondary spermatocyte. Okay. Now, this secondary spermatocyte undergoes meiosis too, releasing 4 haploid gametes, 4 haploid cells. They are called as spermatids. So, meiosis 1, meiosis 2. Okay. Now, these spermatids now undergo one more process called as spermiogenesis. Why? Because they need to look like sperm now, right? Look like sperm. These spermatids now will undergo spermiogenesis to produce the entire spermatozoa or the sperm. Now, understand, students, this spermatozoa right here, the head of it is embedded in the Sertoli cell. So, take the head present inside Sertoli cell. Now, what happens? Now, all the sperm which are now released from the seminiferous tubule, the process of release of sperm from seminiferous tubule is called as permeation. Release from seminiferous tubule, permeation. So, please know the difference between sperm. Any doubt here, students? No doubt, right? No doubt here. So, remember, second is spermatocyte, spermatids, spermatozoa, spermiogenesis. Download these notes. You will understand on your own. You don't have to work a lot here. It's very easy to understand. Okay. We have something called as gonia. We had spermatogonia. We had the spermatogonia. We have the spermatogonia. Now we have the oogonia. The oogonia again is diploid in nature. Now it undergoes a process called as mitosis, increase in number. Now one such oogonia increases in size right now. We call it primary oocyte. Now this primary oocyte now starts to undergo meiosis 1. It starts to undergo meiosis 1, but it does not finish. In fact, it gets arrested. It gets arrested at your suspended at prophase 1 diploidine stage. It is suspended diploidine stage. Now, what is the female child? See, all is embryonic stage, birth, childhood, puberty. Once the female child reaches puberty, this meiosis 1 is finally completed. Finally completed. Now, once meiosis 1 is completed, that will result in the secondary oocyte. Now. Dear students, secondary oocyte and the first polar body after meiosis 1 during puberty. Now, this secondary oocyte now is released during the ovulation. Now, the secondary oocyte now starts to undergo meiosis 2, but again it is arrested. At metaphase 2. Now, when does it get complete? It gets completed during fertilization. During fertilization, when the secondary oocyte comes in contact with the sperm, that finally finishes the meiosis 2. And that will result in the ovum, ooted, or second polar body. So please remember this entire oogenesis, very, very important. Very, very important. Okay. Now, students, during whenever this oogenesis is happening, simultaneously at the same time, there is something called as embryo develop or follicle development. Okay. Now, understand, students. Understand. What is primary follicle, secondary follicle, tertiary follicle, Graafian follicle? Primary follicle has something called as primary oocyte plus layer of granulosa layer. secondary follicle has primary oocyte, more layer of granules. Tertiary follicle what has something called as theca externa, theca interna. It has something called as fluid filled cavity called as antrum, and in tertiary oocyte we have the in tertiary follicle we have the secondary oocyte. Okay, so in tertiary follicle primary oocyte becomes secondary oocyte. Okay, then finally we have the Graafian follicle. When it ruptures, it will release the secondary oocyte. That is your entire oogenesis. Now here students, in entire menstrual cycle, you need to remember something called LH surge. Can anyone tell me, students, when does the LH surge happens? Anyone who can tell me? When does the LH surge happens? LH surge. Anyone? LH surge here happens at the 14th day. Here at the 14th day, LH surge happens. So they can ask you, match the following. Remember students, this follicular phase is happening in the ovary, proliferatory phase is happening in the uterus. Similarly, luteal phase is happening in the ovary, secretory phase is happening in the endometrium. Now here they can ask you which hormones are involved. The main question which I have always noticed they will ask you is, which hormone is responsible for the maintaining the endometrium layer? The same hormone is released by corpus luteum. Anyone who can tell me? Anyone who can tell me, students? Anyone who can tell me which hormone is secreted by the corpus luteum, which also helps in maintaining the endometrium layer? Anyone? Yes, I'm from Karnataka. That is called as progesterone. Students, small 2 minutes break and I'll ask you, how many of you saw yesterday's IPL match? How many of you saw yesterday's IPL match, students? How many of you saw IPL? Last 2 days, from last 2 days, right? I feel the entire South India is very happy. RCB won. Second match, your SRH won. Next level batting SRH. Travis had blasted everything. Then third match, Chennai also won. Right? Next level bowling and batting. Right? So next level it went. LH search done. Opposite progesterone done. Menstrual cycle start. The starting of menstrual cycle. The starting of menstrual cycle is called as menarche, and the ending is called as menopause. Fertilization takes place where? The entire fertilization takes place in the ampulla region. Okay. During fertilization, the sperm comes in contact with the zona pellucida layer and the plasma membrane of the ovum. It induces certain changes and ovum blocks the entry of additional sperm. That is called as fast block method of polyspermy or fusion of multiple sperm. As soon as one sperm comes in contact with the egg, the entire membrane of the ovule, okay, it will become depolarized, so no more sperm can enter inside. That is called as fast block. There is something called as slow block also. That is cortical reactions. You don't have to worry about that. Okay. Friends, I don't think I have to teach you this one, right? I don't think so. I have to teach the entire sex determination. Very easy. I'll do it, Manu. Now, this is very important. This many students leave it out. What is that? That is called as cleavage. So, what exactly happens in cleavage, students? Now, we have zygote here. Zygote will divide into 2 cell, 4 cell, 8 cell, 16 cell, and blastocyst. Eventually, blastocyst. This 2 celled, 4 cell doesn't have a name. While this 8 cell, 16 cell, this area, this entire growth phase is called as morula. This is called as morula stage. Now here students understand, this each cell here is called as blastomere. It is called as what? Blastomere. Individual is called as blastomere. Individual is called as blastomere. Okay. You can see a development happening here. Now once we have the blastomere, see, blastomeres, individual is called as a blastomere. Now once this blastomere develops, right, eventually what happens is, in the blastocyst, all the blastomeres are arranged in two ways. Most of them are in the corner. The corner part is called as trophoblast, which will develop into placenta eventually. Remaining blastomeres will arrange in one corner, that is called as inner cell mass, that will develop into embryo eventually. This is what you need to remember. Okay. Inner cell mass. Now, implantation happens. Implantation is a process in which blastocyst gets embedded in the endometrium of the uterus and lead to pregnancy. After implantation, finger-like projection appear on the trophoblast called as chorionic villi, which are surrounded by uterine tissue and maternal blood. And this chorionic villi plus maternal blood, when they join together, when they become interdigitated, that will result in the formation of something called as placenta. Now, students, very important question for your board. They can ask you, what is the function of placenta? Exchange of nutrients, exchange of oxygen, release of waste, right? Removal of waste. And also, students, remember, placenta actually act as a temporary endocrine gland. The placenta actually acts as a temporary endocrine gland, and placenta can actually produce hormones. It can. The hormones such as HPG, HPL, right, HCG, human chorionic gonadotropin, human placental lactogen, estrogen, progesterone, and also in the later stages it can also produce relaxin also. And relaxin is also produced by, remember, relaxin is also produced by placenta also. It is not given in NCERT properly. Okay. Very important. HCG, HPL, estrogen, progesterone. Okay. Relaxin is also produced. After implantation, inner cell mass differentiates into outer ectoderm, inner endoderm, and middle mesoderm. Ectoderm, endoderm, mesoderm. These three different layers will produce different types of organelles in our body. The inner cell mass contains certain cells called as stem cells, which have the potency to give rise to any other tissue or organ. Very important again, very important. Development of baby, development of bone. Yes. Estrogen is produced by your placenta also. Students, see, relaxin is produced by ovary, true. NCERT line it says that, NCERT line says, in later stages, relaxin is produced also by the ovary. Hit the line properly, students. I'll show you. Where many students go wrong. Students, see this line here? Can all of you see, students? Is that a hormone called as relaxin is also secreted by the ovary. Right? Also secreted by the ovary. Right? There are many times that even relaxin is also produced by placenta also in cases. Right? Don't be confused. Do a small Google search, you will get to know. Is placenta producing relaxin? The answer will be yes, it does produce relaxin. Okay. Very important line, students, read this. By the end of second month, the fetus develops limbs and digits. At the end of the first trimester, most of the organs are formed. The first movement of the fetus and the appearance of the hair of the head is by month. Very important line. They say, right, the baby is kicking. The baby is kicking, they say, right. That is happening in the 5th month. The average duration of human pregnancy is about 9 months, which is about gestation period. The parturition is in, what is parturition? Parturition is nothing but childbirth. That is called as parturition. Right? It is induced by complex neuroendocrine mechanism. The signal for parturition originates from a fully developed fetus and placenta. To ask you a question, Where is the signal for parturition coming from? The signal for parturition is coming from the fetus and the placenta. Now, always remember, students, once the signal comes, right, there will be small contraction, right, which induces mild uterine contraction called a fetal ejection reflex. Now, once this happens, what happens? As soon as the small contraction happens from the pituitary, what happens? Oxytocin is released. Now, once oxytocin is released, what happens is that more contraction, more contraction, more oxytocin, more oxytocin, more contraction. So it keeps on happening until the contractions are enough for the baby to slide out. That is called as parturition. Now, once the baby is born, it needs only mother's milk, called as colostrum, right? Because it is rich in a lot of antibiotics. So always the doctors recommend mother's milk is important in the early stages. Okay. Colostrum, which contains several antibodies. Okay. Oxytocin, hormone acts as the uterine muscles, first strong contraction, that maternal pituitary. Students, I think that is the end of my time right now. I will come back for principles of you will do right now. Okay. Students, do you want reproductive health or not? Do you want or not? Tell me if you IGA exactly. You don't have an option. Okay. So now ma'am will come, students. Ma'am will start. Right. Ecology, I will do it. Ecology will see. It will very easy. Okay. I'll give you questions on ecology. What you need to study. I'll come into principles of nature. So do you remember? Do you realize, students, in just 1 hour 45 minutes, 1 hour 40 minutes, we finish two chapters. Ma'am, and you have to make like this. Hold like this. Otherwise, the voice will break. Pocket, if you put it, you can't turn. I'm writing like this from that time. Can't turn at all. Okay. So I'll come back for principles of written, students. So I have one more class right now. I'll finish that class and I'll come again. Okay. So bye bye, students.
Yes. Now it's better. Pinning it. Okay. Now. Let me know if it's clear. That's fine, right? Right voice. Form them. Okay. Fine, right? Start. Everything is okay. Let's start with reproductive health. reproductive health, I'll not be teaching you every single thing. I'll not be teaching you every single thing of the chapter, but there are some important things that has been repeated in the CBSE in the past years. Those topics I will be. Okay, okay, yes, yes, voice is clear. Okay, reproductive health. First, we are starting with beautiful summary is there. Beautiful summary is there. All the dates are there. Everything is there. All that you have to do is carefully. Okay. So, according to WHO, that is the World Health Organization, reproductive health is still well all aspects of reproduction. That is emotional, and social. That is just by telling that okay, my reproductive system is healthy does not mean you're reproductively. That means you're not healthy. Okay, even when we talk about health in human, we say that health is an aspect of physical, mental, social. But physically you should be fit, mentally also you should be fit, socially also you should be fit, right? That is the reason why rapes happen, because these people want to hurt someone to get the satisfaction. That is not like, yeah, they are healthy. That means what? They are only reproductively active. They do not have any or social, right? So here also students, emotional and social. 2 marks question can come. What is reproductive health, or define reproductive health? Okay, got it? For those who do not know me, I am Miss Gopika. Okay, I don't know how come you be. Okay, it could be because some of you are new CBSE students. Yeah. Okay. Now look here, students. Reproductive health problems and strategies. What exactly happened, or what's the reason for us to even think of why we reproductive health is so important? Because India initiated reproductive health program. Important reproductive health programs, family planning in 1951. Why? Because there was no control in population. Now also there is no control in population. Okay, that is because what is the main reason? Even if I hold the mic tightly, it will not happen. Okay, yes, slightly break. So India initiates reproductive health program in 1951. That's because people were not having any control. Right? There was no control in population. Population kept increasing. Now, students, just because population is Raju from Tamil Nadu has to make that much rice because here in Bangalore population is not necessary. That means even though population will continuously, it is not necessary that the resources increase. Resources will still be stable. Understood? Yes. Clear? Yes, my health is okay now. You can see the mark. I feel mark, no? Yes, it's better. So I thought today I will not wear the mask. Clear? Yes. Voice. How bad is it? Because it was. I've informed them. Just let me know if I pin it down. Like if I pin it here, is it better? Better now? Since last time also this happens. Better? Yeah, no, it's better, right? Not let anything class. Okay, hello. The class. Audible? Fine, better. Okay. So India initiated reproductive. I'll get the mic change. India initiated reproductive health program, that is family planning. Wider reproduction related areas are in operation under the reproductive, and this is very, very important. They can ask you in MCQ. They can ask you as a full form. Reproductive and child health care program. Friends, in this chapter there is less to explain. If you notice, this chapter is all about some terminologies and some things, right? So here you have to understand that reproductive and child health RCH program. Such programs deal with the following. What is the program necessity? First of all, population was increasing. Second of all, people do not know what are the contraceptive methods, how they can control, okay, the population. So this awareness was given by RCH. Okay, remember PYQ question, this is, sorry, MCQ question, this. So remember this carefully. Done, done, done. Okay, very good. Okay, give awareness about reproduction related aspects for creating a reproductively healthy society. Here, this is the main thing. So I've told you the dates. Now again, dates which are very important. Here there's no much to explain, but let's just read and go. Okay, let's just read and go. In 1900s, world population was about 2 billion. How much, students? In 1900. But now we are at 2025. In 1900, the world population, but by 2000, it rocketed to about 6 and 7.2 billion. Can you imagine? Can you imagine? Right? When it was in 900, it was 2 billion. Then it skyrocketed in 2000 to 6 billion and at 7.2 billion. And now here comes the most proud moment of being Indian is here. In India, population was nearly 350 million at the time of independence. It reached 1 billion by 2000. That means, friends, in every 6 person, 1 person would be. Because total population was 6 billion in 2000. Out of that, 1 billion was contributed by India. That means, out of every 6 persons, this, if I see people together, 1 person would be. Are you proud? Are you proud? Out of every 6 person, 1 person in India. Okay. Now moving forward, it crossed 1.2 billion in May 2011. Again, out of 7.2 billion, 1.2 billion is still contributed by India. It means every 6th person in the world is an Indian. According to 2001 census report, our population growth was less than 2%, okay, a rate at which our population could increase rapidly. Now they will ask you the question that what could be the reason population skyrocketed? What will your answer be? You should say it is because of how increased health facilities. Now students, just imagine during your parents' time, during your grandparents, forget about parents, grandparents' time, there was people like, midwives. I don't know what you call this midwives, your language. People who used to do the childbirth, right? Like the labor. The labor was done at home. Okay, at home. Now, because of that, what used to happen? They used to not have facilities. Like if the oxygen goes down or if there is a blood supply, issue, they did not have any facilities. They just knew the technique. They used to come to home and do. Now, during this time, there are chances that out of 13 kids, only maybe 6 survived. So, population was below. But in the growing world, as advancement kept coming, everywhere there started being clinics, hospitals, because of which facilities kept increasing. Here, so reasons for population explosion is increased health facilities. Question can come, please make note, increased health facilities, better living conditions. Do you agree now? Yes or no? We have health insurance, we have lot of hospitals, every single places, whatever kind of treatment you want, there is always places, right? So this has happened, okay? Yes, I don't want the energy to go down, the class is going to be longer, so no energy should be down. Energy shouldn't be down. If you don't want to score well, then fine. Here, okay. Rapid decline in death rate, maternal mortality rate and infant mortality rate. So students, as I told you, during this entire labor time, there are chances that the mother is not well or the child is not feeling well. Because of it, one of their death happens. Because that used to happen continuously, there were chances that, okay, the population will not skyrocket. But as the healthcare facilities kept increasing, IMR and MMR rate decreased. Okay, IMR and MMR rate, because of which also there was an increase in number. Increase in number of people in the reproducible age, that also increased. Okay, people who were at a reproducible age and able to give birth, that age also increased. Here, yes, obviously the new method here, new method is better because there is, in case there is a problem with, let's say, in case there is a problem, there is mic issue, Mithun sir, the complaint. English. Boys, now I'm holding like this, the hand. Okay. Since sir has come, students, put like 2, 3 fire emojis. From 2 days we have issues. Finally, we have got appointment. Hello students, is it better now? Let me know. Yes, please let me know. Is it better? Yes. Better, no? Akshit, wait there. Voice is too better, but voice is too. We're doing that once. Students, how about now? Is there a difference? Is the voice fine? Voice is distorted, my god. Should we change the mic? So sorry students for the problem. Students, how about now? Yeah, better, no? Okay. Oh, fine. Okay, thank you, sir. Okay. Better students, right? Okay, chalo, let's start the class. Okay, there's a slight echo. Let's just continue if it's not that bad. Okay, now this is clear, right? This is clear, right? Now moving forward to intrauterine devices. This is a very important concept from both NEET point of view, from both CBSE point of view, intrauterine devices. What are intrauterine devices, students? All of you know that there are barrier methods, right? Barrier methods means what? Natural methods. Okay, like withdrawal technique, lactational amenorrhea. These are natural methods. Then comes barrier method. Barrier methods, you have all these condoms. Okay, condoms, IUDs, right? We talk about all of this here. Now, in natural method, it's very like what to say. It's just a rhythmic that okay, a person is predicting it. It is not assured. Okay, there is no 90 or 100% chances that the person will not get pregnant. That does not mean that. Okay, natural method is just like a trial and error. Okay, it will not happen because the woman is lactating, or the woman is going through periods, and then the fertile window is after that. So before that, all that is just a calculation. But when you come to barrier method, when you come about IUDs and all are the top, okay, highly used techniques in India. Okay, these are inserted. So students, the problem with intrauterine devices, like how we talk about condom, there is no easy accessibility. Accessibility means you will definitely get it, but you cannot use it by yourself. Like you cannot wear it by yourself. You need a doctor or a nurse who is expert in this to do it. So you will have to insert it with the help of a doctor or nurse in the uterus through the vagina. They increase phagocytosis of sperm. What is phagocytosis? Phagocytosis means engulfing. Okay, engulfing. Have you seen that? Our WBCs also do phagocytosis. They come like this. If they see a foreign particle, they will try to engulf like this. So engulfing is called as phagocytosis. Okay, yes, very good. Now they increase phagocytosis of the sperm. Now students, the most important thing is types of IUDs. Non-mediated IUDs, that means the IUDs that do not have any hormone or any chemical release, these are called as non-mediated IUDs. They retard sperm motility. All of you know sperm has to reach the ovum. So they will stop or they will reduce the sperm motility so that they do not reach to the destination, correct? So they will retard sperm motility, also have spermicidal effect. Okay, have spermicidal effect. Example, Lippy's loop, very important. Very, very important to remember. Lippies loop, non-mediated IUDs. So what do these IUDs do? We have three types: non-mediated IUDs, copper-releasing IUDs, and hormonal IUDs. Non-mediated IUDs means these people do not release any hormones or chemicals, but then definitely they do the job of retarding the sperm motility. But when you come to copper releasing IUDs, they'll be continuously releasing the copper ions to make the difference. That is what, students, copper releasing IUDs are. They suppress the motility and the fertilizing capacity of the sperm. What do they do? Again, suppress the motility. Let's say you're going to some place, I puncture, okay, you're going in, let's say, car. I remove the air and give in the car. Now, will you reach the destination faster? You will not reach the destination faster. Something like that. Here also, we reduce the motility of the sperm, so they do not reach to the, ovum. And students, at the same time, we also reduce their fertilizing capacity. Now, why do we do art technology? Because many of the people do not have good fertilizing capacity. Sperm does not have fertilizing capacity, right? So, this is important, okay? Clear? Now, look here, students. CUT, CU7, multi-load 375. These are the three examples that you need to remember. Now, two examples you will remember easily. Why? Because, ma'am, you told it is copper releasing IUD, so it will be copper 7 and copper T. Multi-load 375. Okay, number, everything has a meaning to it. Okay, it's not like chumma someone gave 375. So, are you all smashing the like button? Let me see how many likes happened. Yeah. Let me see how many likes happened. Yes, we need to make it fast. 300, 400, 500. Faster. Okay. Now, hormone releasing IUDs, they make the uterus unsuitable. Very, very important, students. Very, very important. Hormone releasing IUDs. Hormone releasing IUDs, they make the uterus unsuitable for implantation to happen. Just now you studied implantation. Just now you studied about everything in the uterus. So, it will be easy for you to connect. Okay. It will be easy for you to connect. Okay. Thank you, Stella. So, hormone releasing IUDs, they make the uterus unsuitable for implantation and the cervix hostile to the sperm. So, basically they are making the environment a negative, like setting a bad environment so that the sperm does not survive in that environment. If we provide everything, usually what happens, the uterus or from the vagina as it enters, the body, the female body makes it suitable for the sperm to survive. But here what are we doing? We are doing the opposite of that. Okay, we are doing the opposite for that. Clear? Yes. Okay. Example, progesterone and LNG20. Okay, progesterone and LNG20, students. Always remember, your hormone releasing IUDs can be a combination of two things. What, ma'am? They can be a combination of progesterone plus estrogen. Can you see the blue color? Or later you will tell me, ma'am, who writes in blue color? And a progesterone complex. Okay, just now you studied again the uses of hormones, so these topics will be easy to connect. Progesterone plus estrogen and progesterone complex. So what they are going to do, they are going to make sure that implantation does not happen. At the same time, they do not give the cervix, okay, does not make the cervix hostile for the sperm. Clear? Yes, yes, students, I understand. I totally get it. We are very poor. Okay, you need to make us rich. Okay, yes. Now, here, students, empty piece. Okay, empty piece. Can you see it? Is it too small? No, right? Okay. Yes, very good. Progesterone alone or progesterone plus estrogen complex. Okay. Now, empty piece. What is empty piece? Medical termination, okay, procedure. Medical termination procedure means what, students? Let's say, here you have to understand one thing very importantly, I would say. Holding this mic is one big task. Can I pin it somewhere? Just let me know if it is very horrible if I do that. I'm not able to hold and teach students. My concentration is in this. Okay, how tight I am holding this. Just a minute, just a minute. When this technical issues will get over. Is there a difference? Is there a difference just because I kept it on the this one? Any difference in the voice? Quickly tell. Any issues? Tell fast. Lazy people. No, ma'am, it's fine. Okay. Now look here. MTP, one most important thing that students don't understand is between contraception and MTP. Students, MTP is medical termination. That means pregnancy has already happened. Pregnancy has already happened. Let's say the couple did not know. Okay, couple did not know, let's say after 2 months they got to know. Okay, after 2 months they got to know. Now they have decided that no, we cannot take care of this child, so we need to abort it. Okay, so during that time they go for MTP to a doctor. Then the second scenario is if there is a rape. Okay, if there is a rape and by mistake the woman or the child has been pregnant, then we can go for MTP. Third scenario is if the mother's life or the child life is in danger. Okay, if the baby that is going to be born, the fetus as well as the child, mother has any problem, that time also we can go for MTP. Understood? You can go for MTP. So students understand, 45 to 50 million MTPs are performed in a year. Can you imagine? 45 to 55 million MTPs are performed every year. That is equal to 1/5 of the total number of people who are getting pregnant. Okay, 1/5 of the total number of conceived pregnancy. So MTP helps to decrease the population, but the problem is some people take advantage of it. Right, there are a lot of couples who do intercourse without using contraceptive methods, and for that also they go to MTPs and they go to unqualified people. That means, you know, people who just open the clinic and just get the doctorate from some random place, and that kind of people do it. And because of that, what happens, students, like in the cases like ectopic pregnancy, or then cases where, you know, there are some cells, like let's say they would have given a pill, there are some cells still in the body, that cells will later become infections. That kind of scenarios has happened where people have lost their life also. So it is very important that when we do such kind of techniques, we have to go to a proper doctor who has qualified it. Okay, now many countries have not legalized, you know this, no, MTP is not legalized in many countries, but again government of India legalized it in 1971. Okay, with some serious strict conditions. Now students, again I told you when MTP has to be safe. Okay, this is a very important question. MTP are safe during the first trimester, that is up to 12 weeks of pregnancy. Up to 12 weeks of pregnancy you can do MTP, that is you can go for you know, abortion, but after that the abortion is very risky. Okay, after that the abortion is very risky. Got it? Now students, majority of MTPs are performed by illegal method, that is many people perform it without even having proper, you know, knowledge about it. They just give a pill and they'll tell you take this pill, you will be fine, but you are not supposed to do it. Now tell me students, what are the 3 scenarios when a person can go for MTP? First I told you, if the couple is decided that they do not want to have a child right now, they are not ready for it, they can go for MTP. Then the second scenario is people who have like rape has happened. Third scenario is if the mother or the child is going to be in danger, or the child has a genetic disorder which cannot be rectified, or the child is not breathing, like in the sense heartbeat is not there, any of the organ failure has happened, and they are able to understand with the scan, they will tell okay you can go for MTP. Got it? Clear to all of you? Yes? Yes, very good, excellent. Now amniocentesis students, amniocentesis is very important to understand. Why? Because amniocentesis happens, like amniocentesis is done by taking few cells of your amniotic fluid. Okay, so in this, some of the amniotic fluid of the fetus is taken to analyze the fetal cells and dissolve substance. Now students, what happened with amniocentesis is, it was a good thing because you could understand if the child is going through any genetic disorder. But when it became a bad thing, when people started using amniocentesis to do female feticide. Female feticide means what? That okay ma'am, I got to know that the child is girl child, so I am going to murder. Okay, I am going to kill the girl child and I am going to abort the girl child. So during this situation, what happened? We noticed, the government noticed that there is a continuous increase in female feticide and we need to do something about this. Okay, we need to do something about this. So because of which amniocentesis, that is amniocentesis is not banned, amniocentesis is still there in India. The sex determination part is banned in India. That is why we do not have any gender reveal. If you notice, foreign everyone does gender reveal, right? Every alternative day they will be doing gender reveal, it will be coming on YouTube. But in India, gender reveal, that is sex determination part is not allowed. Amniocentesis is still happening in India, just that sex determination part, that if the child is boy or if the child is girl, that is not allowed. Clear to all of you? Yes, yes, anything, sickle cell anemia, your Turner syndrome, Klinefelter syndrome, any kind of diseases like the thalassemia, hemophilia, everything we can detect it with amniocentesis. Okay, so this is important. So look here students, the Medical Termination of Pregnancy Amendment Act in 2017 to reduce illegal abortion and consequent maternal mortality and According to this act, a pregnancy can only happen in the first trimester, that is up to 12 weeks. After that, it is very risky. Okay, after that, it is very, very risky. Is this clear to all of you? Yes. Now, next is art technology. So, I am only talking from exam point of view, what you have to study at this last minute. So, MTP, aram se you can write 5 marks question about it, you can write 3 marks also, you can write about 2 marks also. Because you start with what is MTP, you start with when a person can do MTP, what are the 3 conditions when we do MTP, was it legalized or is it not legalized, and then 45 to 50 million happens every year, it is only 1/5 of the total pregnancy that is conceived. Clear to all of you? This much you can aram se write. Okay, quickly smash the like button, let's make it 300 likes very fast. Now, it is the ability to conceive or produce children even after 2 years of unprotected sex. Okay, that is, students, even after 2 years of unprotected sexual or intercourse. Okay, sexual intercourse, if the couple is not able to conceive, okay, sexual cohabitation, the reason for this may be physical or congenital. Okay, sometimes it can be because of some kind of drugs that some people take. It could be because of, just give me a minute, students. 1 second, just. Quickly smash the like button, all of you, don't be confused. Yes, okay, done. Yes, so here 2 years of unprotected, and this could be because of some immunological problem, could be age, could be genetic disorder, could be food habit, could be lifestyle, many reasons like that. Okay, now assisted reproductive technology or the art technology. Okay, what is this art technology, students? That is where we give the same setup, right? Same setup means what? The same in vivo conditions, whatever is there inside a female's body, we give that same kind of condition in the lab and we do. And art technology is very common nowadays. All of you know this, no? Art technology is very common nowadays. All of you would have watched that movie called Good News. In that Good News, they are clearly telling what is art technology. They are showing how embryo transfer happens. So everything is very clear nowadays. So many IVF clinics every single corner. So in vitro fertilization, that is IVV. Students, in vitro, okay? In vitro means outside. In vivo means inside the body. So outside means where? In the lab. Okay, in the lab. So in vitro fertilization or test tube baby program, in this method, ova, important. In this method, the ova from the wife or the donor and the sperm from the husband or the donor is taken. They are collected and induced to form a zygote. So what are we taking? We are taking a petri plate, we are taking a same setup, we are taking ova from this woman, we are taking a sperm from this person, and we are doing it inside the lab. Okay, we are doing it inside the lab, correct? So under stimulated condition, this is followed by embryo transfer. After the embryo is formed, we will transfer this into the fallopian tube of the female, or if we are seeing that okay, the fallopian tube is not possible, then we do different kind of techniques. Okay, clear to all of you? Yes, I do not want you people to text each other in the chat box. This is not your WhatsApp channel. Okay, this is not your WhatsApp chat. So if you're going to talk like this, I'm going to remove you from the class. Clear? Yes. Fine. Okay. Now, listen to me carefully. Listen. So this is followed by embryo transfer. Now, students, embryo transfer is of 2 types. Okay, embryo transfer is of 2 types. What is the 2 types of embryo transfer? Look here. First is zygote intrafallopian transfer. First is zygote intrafallopian transfer, which is ZIFT. What is it? ZIFT. Okay, clear to all of you? Okay. Now, ZIFT, that is zygote intrafallopian transfer, transfer of zygote or early embryo, this is very, very, very important. Very, very important. Okay. Yes, zygote intrafallopian transfer is transfer of zygote or early embryo within 8 blastomeres. 8 blastomeres. Question will come. Up to 8 blastomere stage. This is up to 8. This is more than fallopian tube was healthy, no problem was there, nothing twisted, not the narrower, nothing problem was there, so we transferred it into the fallopian tube. Another scenario, we saw that fallopian tube, it's not possible, so let's do it into the uterus. So we did intrauterine transfer. That is more than 8 blastomere stage. Okay, more than 8 blastomere stage. Now students, embryo formed in vivo fertilization. That means what? Fertilization within the female. In vivo. That means inside. Okay, in vivo, inside. Inside. Okay, fertilization is also used for such transfer to assist those females who cannot conceive. Okay, assist those females who cannot conceive. Okay, there is no lag. Okay, now students, gamete intrafallopian transfer. So in vivo we have two techniques. Gamete intrafallopian transfer, that is transfer of ovum. We transfer the ovum from a donor into the fallopian tube, okay, of another female who cannot produce the ovum but can provide suitable environment. Now we have learned about surrogacy, no? Surrogacy also what happens, students? What is exactly happening, students? Is there a lag? No, right? No lag, right? Chumma, people don't tell. Please keep responding in the chat as you are understanding. Please put thumbs up, otherwise I will not know, you know, if you are understanding. If there is a lag, I will not understand. Okay, so please put thumbs up as you understand. Okay, yes. So here what is happening, students? In surrogacy also what happens? Ovum and sperm will be given from the mother, like from the, let's say from two stars, two Bollywood stars, one is giving ovum, one is giving sperm, but the surrogacy, that means another woman's, the womb will be used for the baby's growth. Now this happens, why? It could be because of they are not healthy enough to carry the baby, all of that. Here what is happening, we are taking the we are taking the egg from another person and we are injecting it into the fallopian tube, but the other person, let's take example students, let's take X is there, Y is there. She cannot provide the ovum, so we cannot get ovum from her. So what we will do, we will take ovum from Y. Okay, we will take ovum from Y and we'll add it into her, inject it into her fallopian tube and she will provide the environment. Understood? She will provide the environment. Is this clear? This also happens. Okay, this is called as gamete intrafallopian transfer. Gamete intrafallopian transfer. Next is intracytoplasmic sperm injection. It is a laboratory procedure in which a single sperm, okay, we take a sperm from the male. This could be because students, the low sperm count. Okay, the males do have very low sperm count. Fertilizing capacity is comparatively less. In these scenarios, what we do, we'll take from the, we'll take the sperm, we'll directly inject it into the cytoplasm. So it is a laboratory procedure in which a single sperm is directly injected into the egg. Usually what happens, just now you have studied in reproduction chapter, that the sperm has to go and fertilize the egg, right, by going through the layers. So we notice that the sperm is not able to do that because there is low sperm count or the fertilizing capacity is too less. So in that scenario, we'll take the sperm, add it into the cytoplasm of the ovum. Done, 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 done. Last technique of this chapter, artificial insemination. Okay, so after fertilization the embryo will be implanted into the woman's uterus. Okay, clear? Artificial insemination means, students, the semen collected from the husband or donor is artificially introduced into the vagina. So we have taken the semen, we have collected it and we are introducing into the vagina. That means we are not, or to the uterus. Understood? Like we are not injecting it directly into the cytoplasm, we are not making embryo and giving it, we are not making zygote and giving it. What we are doing? We are telling sperm, you only travel, we will send you for the fertilization. Okay, so if the male is not able to, that the sperm is not able to travel all the way from vagina to the uterus or even cross, okay, that time we do artificial insemination. That is, we take it artificially, we introduce it into the vagina or uterus. Artificial insemination into the uterus is known as intrauterine insemination. Now many of you will get confused with IUT and IUI. This you please understand, this we study under in vitro embryo transfer. Okay, embryo transfer up to more than 8 blastomere stage. This is transfer, this is insemination. Okay, this we study under AI. Okay, artificial insemination. So artificial insemination into uterus is known as intrauterine insemination. This technique is useful for male partners having inability to inseminate female, or they have a low sperm count. Understood? Or they have a low sperm count. In good news, they are telling one thing, no students, when Akshay Kumar's sperm is going, right? They say that no, when Akshay Kumar, like the sperm is traveling, it becomes unconscious in her world. That means what they're trying to say, they're trying to say that the sperm is not able to reach and fertilize, and upon that, because of age, they also have a low sperm count. That is why they're going for IVF. Understood? Clear or not? Chapter is clear. Can I go to the next chapter? Can I go to the next biggest chapter? Molecular basis. Hmm? Possible? One second, students. I'll just wipe. I don't have my bag. Shall I just wear the glasses and come? Can you all wait for a minute? Just 1 minute. I'll just wear my chashma. Do you guys want a break? PDF will be available on the Telegram channel, on this Telegram channel, as well as Vedantu NEET English. Do you want a break? 5 minutes break you want? Because you people were sitting for 2 hours. No, I just came now. Just let me know if you want a break. I'll give you a break. I don't have problem. Let's take break only and do the classes, because otherwise it'll be too draining. Because again, after this, I have to do biotechnology, evolution, principles, application. So that's why. Now we'll take a break, and then again we'll take a lunch break. So 5 minutes break is enough, students. Not more than that. Okay, you be here only. Come back at 12:40. Nothing more than that. Like not big breaks. Just 5 minutes break. Okay, just for you to refresh and come. Like Yes, are you all here? Oh, ready, ready? Finish here only, no? Come on, let's continue. Let's continue. All your friends, ask them to come and join. Ask them to come and join. Yes, let's start. Start. Okay, molecular basis of inheritance. My God, one big chapter this is, right? So students, molecular basis of inheritance also we will do from exam point of view, because you know, if I take this chapter, I at least need 5 hours to complete this chapter by going into the details. So here what we will do, we will do it from the surface level, but at the same time you will be getting a clarity on the topic, because you are not studying it for the first time obviously, and you will also be able to perform really well in your exam, because you will know what to write and what not to write. Okay, clear? Because evening anyway we are doing the question. Principles of inheritance will be taken up by Basavaraj sir. Right now Basavaraj sir is in a batch. After he comes, he'll take principles of inheritance. Okay, we'll be going in a little random order because we both have different timings classes. Okay, yes, ready all of you? Yes, chalo, let's start. Molecular basis of inheritance. So I have already given my handwritten notes. It's already available on the Telegram channel. Okay, but here we are going to learn with the notes. Students, all of you know about the nucleic acid, right? That there are two types of nucleic acid, DNA and RNA, and this was found by Frederick Misha, and later it was named by Altman. But this and all won't come in your examination. What will come in your examination is that they will ask you about your nitrogenous base or the structure of your polynucleotide chain. Now, when you think of polynucleotide chain, what are you supposed to think of? You are supposed to think about your nitrogenous base, sugar and phosphate, right? You have to think about your nitrogenous base, sugar and phosphate, and they are bonding. Okay, so first we will finish this slide and we will come into the next slide. Okay, so students, look here. What is your nucleotides made up of? Polynucleotide. That means what is your DNA and RNA made up of? You should know that these people are polynucleotide. Polynucleotide means what, ma'am? Many nucleotides together. But ma'am, you didn't tell us what is nucleotide only. Yes, what is nucleotide? So students, there are 3 components to a nucleotide. All of you tell me in the chat box, and you have to make the target 300 likes. Come on. Yes, what is nucleotide? Ma'am, we know that they have sugar as one component. We know that one person has nitrogenous base is one of the components. Nitrogenous base, and the other component is phosphate group. This also all of you know. This is phosphate group, correct? Yes. Now students, they are telling us that sugar is of 2 types. Depending if it is RNA or DNA, we have 2 types of sugar. What are they? It is called as ribose sugar and deoxyribose sugar. Deoxyribose sugar, correct? Now, ribose sugar means they have one presence of one OH, okay, in the second carbon. In the second carbon, they have an OH extra. They have an OH extra. But in deoxy, that OH is missing. So, in the second carbon, they just have an H. They just have a H. Understood? This is the major difference. Now, ma'am, does that make such a big difference? Yes, it do make a lot of difference. This is the reason why your DNA is one of the most chemically and structurally stable structure. Chemically and structurally stable. Clear to all of you? The reason for this to be chemically and structurally stable is because of the presence of that, lack of that O in the second carbon. Because if oxygen is there, it will go and bind with everyone. It is so highly reactive. It will be like, okay, I want to be with you, I want to be with you. So it will just go everywhere and it will react. So to out, because that oxygen is not there, okay, we have one advantage. We have one advantage. Clear to all of you? Yes or no? Done, done, done. Okay, so this is what your nucleotide is. Now, many nucleotides, okay, let's consider this one whole unit is there. Many nucleotides together will form your polynucleotide. Okay, polynucleotide. and this ultimately forms your nucleic acid. This ultimately forms your nucleic acid. Now students, it's very important for you to know, now that you know what is there in the sugar, you should know that there are two types of nitrogen bases. What are they? Purines and pyrimidines. Okay, purines and pyrimidines. Now this purines and pyrimidines is not simple. Why? Because purines and pyrimidines have much more to it. Okay, much more to it. I like the Josh. Very nice. Now students, look here. Sorry. Yes. Here, if you see purines and pyrimidines, you will notice that purines are, see students, we tell no, name is small, but their pravarti, what you tell here, their actions are bigger. Same way here also, name is small, purines, but it is bigger, 9-membered ring. Name is big, but very small. Okay, 6-membered ring. Remember like this, okay? Purines are 9-membered ring, pyrimidines are 6-membered ring. Now, one important question that can come, in position 1, 3, 7 and 9, all odd numbers, there is a nitrogen group that is present. That means what students? In purines, okay, in purines, we have to understand that in 9-membered ring, okay, there is a 9-membered ring, and in this 9-membered ring, 1, 3, 7, 9, there is a nitrogen group. 1, 3, 7, 9, there is a nitrogen group. Clear? Now, pyrimidines, name is longer but ring is small, 6-membered ring. Two places nitrogen groups are present, 1 and 3. Okay, 1 and 3. Now, how do I remember, ma'am, what is purines, what is pyrimidines? Purines are pure as gold. Pure as gold. Okay, that is P for pure, A for adenine, G for guanine. P for pure, A for as, okay, adenine, G for gold, pure as gold. Here, now ma'am, how do I remember pyrimidine? Pyrimidine, you remember cut, C-U-T. C for cytosine, U for uracil, T for thymine. So it means cytosine, uracil and thymine. Clear? Pure as gold. Okay, pure as gold and cut is your pyrimidine. Now, cytosine, question again. Cytosine is present in RNA and DNA. Cytosine has no problem with anyone. But then there is problem with two people. Uracil is like, hey, you know what? I will only be in RNA. I cannot be there. I will replace thymine and I will be there. We were like, okay, fine. If you want to do that, fine. We are okay with that. Then thymine also will tell the same thing. Telling that, you know what? I will replace uracil in the DNA. Let this fellow do whatever he wants with RNA. So this is where the complication comes, that uracil is present in RNA, thymine is present in DNA, and these both are getting replaced. Replace means what, students? Let's say this is a DNA. This is a DNA. Let's say we have ATG. Okay, same thing in RNA will be what? AUG. Understood? This is what I meant. Okay, this is what I meant. Did you all understand? As you understand, what are you supposed to do? You are supposed to put thumbs up. Okay, every time I should ask, every time I should ask, every 5 minutes, did you understand? Now, very important, about your bonding. Okay, about your bonding. Now, students, look here. Whenever there is a nitrogenous base plus sugar, now I have told you this multiple times, that in a trio, like in a friends, like let's say a group of 3 people, there's always 2 people who are very closer. Why? It could be because, you know, the other person does not have time or something. So, in a trio, there is always 2 people who have similar liking, similar style. So, same thing here also. In a trio of your phosphate group, sugar and nitrogenous base, 2 people will combine and push one person to the side. So, whenever nitrogenous base and sugar is together, we call it nucleoside, and the suffix for it is "-ine". "-ine". So, what do we write? So, we write adenosine, okay, adenosine, right? So, this becomes what? A nucleoside. Side means what? I'm pushing one person to the side, telling that, hey, you go to the side. Who are we pushing to one side? Phosphate group. Right? Understood? So, in a trio, there is a duo. In a trio, there is a duo. So, the trio is phosphate group, nitrogenous base and sugar together forming nucleotide. In that, one person is going apart. Phosphate is going apart, so it becomes nucleoside. And the suffix is "-ine". Okay, nitrogenous base, sugar and phosphate together is called nucleotide, or else some people can argue, now it's me, human is telling, no ma'am, how can you say that? I want to put it nucleoside monophosphate. I will tell no problem with that, because if this is a nucleoside and if I add one phosphate to it, becomes monophosphate, okay, biphosphate, triphosphate. So I can keep continuing this process, right? Now, talking about bonding. Very, very important. First, let's talk about the duo. That what are these two people bonding together? So, students, whenever a sugar and a nitrogenous base are bonding together, they will form an N-glycosidic linkage. What linkage? N-glycosidic linkage. How do I remember this, ma'am? I'll tell you a very, maybe I don't know if this is even a thing to remember. But see, sugar means glucose, sucrose. We know that, no? Sugar when we tell about. So, sugar you remember glue. Okay, glycosidic linkage. Okay, glycosidic linkage. So, N-glycosidic linkage. Clear? Right? Between, so in a nucleoside, how does the nucleoside bind? With a N-glycosidic linkage. Now, phosphate is like, please involve me also into it. So, phosphate will come. So, when phosphate come, we will tell, okay, fine, you also want to join. You do one thing, you go and join with the 5' carbon of the sugar. So, that 5' carbon will combine with the phosphate to form phosphoester bond. What bond? Phosphoester bond, not diester, ester. Phosphoester bond. Clear to all of you? Yes or no? Any doubts in this? So, now what we have learned so far is that there is a sugar molecule. Okay, I am just drawing sugar. I think I will draw sugar as a box. Okay, this is your sugar molecule. To the sugar molecule, first your nitrogen base will come and bind. NB will come and bind, and they will bind with the help of what bond? All of you tell me in the charts. Charts have to be moving continuously. Then to another side, your phosphate will come and bind. To another side. So, to the first carbon, remember this also. To the first carbon, your nitrogenous base, and to the fifth carbon, your phosphate will bind. So, the first binding, okay, for the first binding, it is going to be N-glycosidic linkage. Okay, N-glycosidic linkage. Okay, linkage. Now, how will phosphate bind? Ma'am, phosphate will bind with phosphate, no? So, remember, phosphoester bond, phosphoester bond. Now, you will ask me, ma'am, but I want two nucleosides to bind together. I'll be like, okay, no problem. So, let's consider this whole thing as one circle. This whole thing is one circle. Now, I want this one circle to go and bind with another circle. Go and bind with another circle, that is another nucleoside. Now, ma'am, what is the bond that is going to happen? So, students, when one nucleoside and another nucleoside bind together, okay, bind together, it will form a phosphodiester bond. Okay, diester bond. Phosphodiester bond. Clear? Yes, very good, excellent. Phosphodiester bond. Clear? So, when one nucleotide and another nucleotide combine, they will form phosphodiester bond. This is a dinucleotide. This is a dinucleotide. So, from exam point of view, your bonding is understood. Okay, bonding is understood. Now, continuing. Salient features. I have seen a lot of questions on this. Salient features of DNA. Backbone of DNA is made up of sugar and phosphate. All of you know this, right? If you understand, you should remember that this nitrogenous base are going to project inwards. So, what is this backbone made up of? Sugar and phosphate, and the base is projecting inwards. That should be your first line of your salient features. Second line is that DNA is a double helical structure which was found by Watson, Crick, Wilkins and Rosalind Franklin. So, when they were trying to understand the DNA structure, they did an X-ray diffraction, and in that X-ray diffraction, they understood that DNA is a double helical structure. Actually, all four of them came to a conclusion at the same time, but three of them got three of them got recognized. Okay, so DNA is a double helical structure. It is in a right-handed fashion. Okay, remember these words, right-handed fashion, and remember, students, purines and pyrimidines, the ratio of purines and pyrimidines should be equal, which is your Chargaff rule. Okay, your Chargaff rule. Purines and pyrimidines have to be, the ratio has to be equal, right? Because if I want to do pairing, if I want to do bonding, how will I do the bonding? So, students, the bonding is going to happen at Gopika ma'am's class, right? The bonding will not happen at Gopika ma'am's class. The trick is at Gopika ma'am's class. That means A will always bind with thymine, that is adenine will always bind with thymine with 2 hydrogen bonds, and guanine will always bind with cytosine with 3 hydrogen bonds. So, at Gopika ma'am's class, okay? Yes, at Gopika ma'am's class. Now students, remember here, this you understood, no? They have anti-parallel polarity. That also you understood, no? 5 dash to 3 dash, 3 dash to 5 dash, they have anti-parallel polarity. Now one turn has 10 base pairs and pitch of the helix is 3.4 nanometer. 2.2 meter is approximate length of your DNA. Can you all write this? Possible to write all of this? Yes or no? It's easy, no? Backbone of DNA is made up of sugar and phosphate and the bases are projecting inwards. The DNA is a double helical structure which was found by Watson, Crick, Wilkins and Rosalind Franklin. Okay, Watson, Crick, Wilkins and Rosalind Franklin. Right-handed fashion, right-handed fashion it is. Adenine will always bind with thymine and guanine will always bind with cytosine. They have anti-parallel polarity. Can you make a note of this? Possible? Possible? Okay, done. And one turn has 10 base pairs. Pitch of the helix is 3.4 nanometers. 2.2 meter long the DNA is. Okay, 2.2 meter long the DNA is. Okay, moving to the packaging of the DNA. So, since I already told you that the DNA is a 2.2 meter long DNA, now if I have to put it into a nucleus which is 10 to the power minus 6, will that be possible? Will that be possible? No ma'am, it will not be possible. Let's take an example into this, what is this? What is this called, students? Glass, okay, glass cover. Okay, to this glass cover, I am telling you, fold your bedsheet and keep. Will you be able to fold your bedsheet and keep? You will look at me and you will be like, ma'am, are you normal? Okay, taking multiple classes, what happened to you? The same thing will happen here also, right? If I fold this, like if I tell that fold a bedsheet and keep inside this, you will be like, you will think I am crazy. So here also same thing, students, a 2.2 meter long DNA, if I have to fit it into a small nucleus, I had to take help from outside. That is, I took the help of a protein which is called the histone proteins. What is the protein name? Histone proteins. So that histone proteins came and they did not come as a single, they came 8 in number, that is H2A, H2B, H3 and H4, double, double, double. Okay, double, double, double. Okay, so here histone octamer. Okay, it formed a histone octamer which is made up of around, okay, it is made up of 8 of them, right? What is the 8 of them, students? H2A, H2B, H3, H4, same replicas again. So it made a octamer. Now this histone octamer will wound around. The DNA is negatively charged, histone is positively charged. So they will wound around and they will form something called as a nucleosome. So histone octamer plus DNA, histone octamer plus DNA is together called your nucleosome. Clear? There will be 200 base pairs. Okay, 200 base pairs in one nucleosome. Now students, to keep all of this in position, we have someone called as H1 protein or linker protein. What is it called? H1 protein or linker protein, because just imagine that there is like this octamer, okay, there is like this octamer, obviously there is wounding of the DNA, right, there is wounding of the DNA, this DNA will go around like this, we need someone to keep it in place, no? So for that we have one H1 protein which is a linker protein. Clear? Yes. So under the microscope, okay, under the microscope, see, no, it is there in prokaryotes also. In prokaryotes we call that entire thing as a nucleoid. We call that entire thing as a nucleoid. They also have the proteins. Okay, we call that entire thing as a nucleoid because it's not scattered everywhere. Now students, remember that lysine and arginine are two basic amino acids. Okay, protein is made up of what? Amino acids. Okay, so the enriched amino acid is lysine and arginine. Okay, now they will condense, they will go through many condensation process and finally form your chromosome. Now you study in principles of inheritance chromosomes, but how did this DNA become chromosome? It is because packaging of DNA happened. That is, we took a protein which is a positively charged protein and we told this positively charged protein, do one thing, let's do one thing. DNA is negatively charged, you're positively charged, let's bound together. So histone told, okay, I will not come alone, I will come with 4 people and we are going to become double and we are going to make an octamer. So when the DNA wounds around the histone octamer, it forms a nucleosome. Many nucleosomes together, they will go through condensation and finally form the chromatin fibers and form the chromosomes. Clear to all of you? Chromosomes. Okay. Now this is what you see in your metaphase, anaphase. When you are seeing under the microscope, you will notice the chromosomes. Now students, in eukaryotes, under the microscope it will look like a bead on a string structure. Okay. Now in what happens in eukaryotes, because higher organisms, we need more packing. No? So for that we will take non-histone chromosomal protein, which is called as NHC. Again, packaging only they will do for higher coiling. Okay, for higher coiling, these people are additional set of proteins. Like, you know, we'll be like, okay, packaging is not getting over, should we bring someone else? So we will bring some additional people, no? That additional people is your NHC. Clear to all of you? Any doubts? Any doubts? Done, done, done. Done, done, done. Can I go to the next topic? Okay. Now, euchromatin and heterochromatin. Euchromatin means, students, euchromatin, okay, loosely packed, okay, lightly stained and transcriptionally active. So, what is happening here? What is happening here? Euchromatin, they are loosely packed, okay, lightly stained. Now, students, just imagine I take one classroom, okay, and I call all of you to play Holi. Okay, I call all of you to play Holi. Now, let's take example, Gayatri is here, okay, Sahil is here, okay, let's say Madhumita is here, Tanuja is here, okay, some people are here, okay, all these people are here, okay. Now, when they are all standing in a big room, only 5 people are there, Now, if I pour some color on them, what will happen? Everyone will not get darkly stained. Why? Because they have lot of places to run. So, because of which they will not, all of them will not get stained. But let's take another example. I take a same room and I fill it with 50 people. Now, if I pour color, everyone is going to get nicely colored because they do not have lot of place to move, and I am pouring colors, right? So, here also same thing is going to happen, students. Euchromatin means these people are loosely packed. So, if they are loosely packed, obviously they are going to be lightly stained, but these people are transcriptionally active. Okay. Now, heterochromatin, tightly packed, darkly stained, transcriptionally inactive. Understood? Transcriptionally inactive. Now, central dogma, this is also a very important question for your CBSE. Okay, very important question for your CBSE. They will ask you directly what is central dogma. Central dogma is like, you know, how in your school you have like a full system, no? Like that in our body also there is a proper system, and on that system we study our molecular biology, okay, genetics, everything is coming under this. Now, DNA can become mRNA, mRNA can become protein, but when DNA wants to like form its own, that means to form its replica, it will do the process called as replication. DNA to mRNA is called transcription. Something is getting transcribed, okay. mRNA to protein, we are translating, the genetic code is getting translated and we are forming amino acids. That amino acids is ultimately forming the polypeptide chain, correct. So, central dogma means DNA to mRNA, mRNA to protein, DNA will form its own replica, that is called as replication. DNA to mRNA is called transcription, mRNA to protein is called translation, correct. Clear to all of you? Yes or no? Understood? Now, students, reverse transcription also is possible, like in your virus, okay. Reverse transcription also is possible in your virus, that is mRNA to DNA, it will be called as reverse transcription, okay. Transcription is DNA to mRNA. Now, reverse means what? mRNA to RNA, DNA, right? Yes? Done? Clear? Done. Pakka? No doubts? Who is taking care of the target? Someone should be there, no, who takes care of the target. Take care of the target quickly. Yes, HIV virus. Very good. Very good. Chalo. Let's, sorry. Okay. Now, students, experiments are all easy. Experiments are, oh, 1 second. I think I need to, okay, 1 second. Okay. Experiments are all easy. Okay. But I will just give you an overview of the experiment. Okay. Overview of the experiment. Is it too bright? Is it fine? Okay. Now, Griffith's experiment, students. Griffith was the person who was doing some experiment. He did not know what he concluded, but he called it as the transforming principle. So, Griffith was working on Streptococcus pneumoniae, which you are going to study in your human health and diseases, the bacteria that causes pneumonia, Haemophilus influenzae or Streptococcus pneumoniae. So he used two strains. One was a smooth strain which was virulent. Virulent means causing diseases. Everything is on the slide only, okay? And then rough strain, that does not cause disease. 2-1 was there. Okay, 2-1 was there. So one was smooth strain, right? Virulent. All of you have to give us lot of energy. Students, today we have batch class, we have KCT class, we have your class. Again, we have class in the evening for most important questions. So today I have almost like 4 classes which are long classes. So if you people don't give motivation, then I will not be having the energy till the evening. Okay, I came at 9:30 in the morning. I will leave the office at 9:30. So I want that 9:30 to 9:30 full motivation from your side. Okay, so yes, keep the energy high. You people are sitting at the comfort of your home. I am standing here and teaching. So I want all of you to have a lot of energy. Clear? Done? Okay. Now, observation. So I'll directly cut through the process, okay, because you know that when he used S strain, the mouse obviously died because it was disease causing. Okay, mouse spelling I don't know, students. Mouse, okay, sorry, mouse died. Okay, then here what happened? The mouse was, okay, mouse lived. But here what happened? He did heat killed S strain and R strain. When he used heat killed S strain and R strain individually, like when he used only this fellow, the mice was alive. Okay, mice lived. When he used this fellow also individually, the mice lived. But the combination of them, the mice died. So then he was like, oh my god, what just happened? I just used 2 things. Okay, I just used 2 things that were keeping the mouse alive, but suddenly when I mixed these 2, suddenly the mice died. So that is when he came to a conclusion that let me call this the transforming principle, because I assure, okay, he was sure, he was not confused. He was sure that something, okay, something from the S strain, got transformed to the R strain. Something from the S strain got transformed to the R strain that made R strain virulent. He was sure of it. So he called that something as the transforming principle. He called that something as the transforming principle. Dun dun dun. Dun dun dun. Yes or no? Clear? Yes, very good. No, R strain was not transformed to S strain. S strain was something from S strain got transformed to the R strain, making R strain virulent. Clear? Okay, good job. Moving forward. Avery, MacLeod and McCarthy's experiment. I saw one question, okay, but not that much, okay. Yes. No, students, something from the heat killed S strain got transformed to the R strain, not ulta. Something from the heat killed S strain got transformed to the R strain that made R strain virulent, because this fellow is already virulent. Okay, so we call that, okay, we call that as the transforming principle. Okay, yes, Stella. Okay, now Avery, MacLeod and McCarthy's experiment is little confusing because it's not given neatly in the NCERT. Now I'll just tell you what you need to remember. See students, Avery, MacLeod and McCarthy's experiment was on the biochemical nature of the transforming principle, because Griffith told that okay, something got transformed, but people wanted to know what got transformed, what exactly, was it a protein, was it an RNA, was it a DNA? Now you know it is a DNA, but that time they were trying to figure it out, right? So he was like, they were like, okay, let's do one experiment, guys, let's do one experiment. So first they took 3 enzymes. What are the 3 enzymes? RNase, protease and DNase. They took 3 enzymes, and then they were like, what are these enzymes going to do? Protease is going to degrade the protein, RNase is going to degrade the RNA, DNA is going to degrade the DNA. So we can kind of understand who got transformed. So first he started with RNase, and when he used RNase, obviously RNA got degraded, not the DNA. So the mice is still going to die. What combination did he take? He took that heat killed plus R strain. Okay, so okay, mice is usually used in the experiment because it's a like, you know, it's like a transgenic animal. That means we manipulate their genes and we work on it. Okay, mice, pig, cattle, we cannot take bigger organisms, so we usually, in all the older experiments, we used to use mice. Okay, So RNAs we took, students, transformation happened. That means what? The mice still died because DNA is still there. Now again we took protease and we degraded the protein. Here also transformation happened. That means again the mice died. But when we use DNAs, what happened, students? There was no transformation. That means the mice was alive. So that gives us a conclusion that yes, the transformed fellow was DNA. But many people were like, no, your experiment does not make sense. Your experiment is not up to the mark. So let's just, like some of them accepted, some of them did not accept. So that is when Hershey-Chase experiment came into place. Okay, what happened, students? Hershey and Chase experiment came into place. Did you understand so far? Yes. Okay, Hershey and Chase experiment in 1952, it was the proof that DNA is the genetic material. So he used bacteriophage, that is the virus that infects bacteria, with a radioactive label. Why, ma'am, they use radioactive labeling, students? Otherwise, when you do centrifugation and when you want to find the ultimate result, you will not know the difference. To differentiate, okay, to differentiate, we use two labelings. What were the two labelings? Okay, to differentiate. Two labelings were that we used radioactive sulfur, which was S34, which was protein labeled. Ma'am, why did you take sulfur 35? Because sulfur is not present in DNA. It is only present in protein. So if I want to label protein, if I want to see if protein is the genetic material, obviously I will take something that is only present in protein. So S35 was present in protein. On the other hand, we took phosphor 32, which was only present in DNA. So problem solved. So if I get P32, it is confirmed that it is DNA, and if I get S35, it is confirmed that it is protein. Yes, uni-equal proof of that Hershey and Chase proved that DNA is a genetic material. Then students, radioactive phosphorus was taken. This also you understood, right? Okay, this you understood. Now, moving forward, DNA label. After infection in the bacteria, only radioactivity, okay, only radioactivity entered the bacterial cells. Okay, only DNA entered, only radioactive DNA entered the bacterial cell, but not the protein. So, conclusion was DNA is the genetic material. Now, ma'am, what if they ask me for 5 marks and I have to write? You write the first step of this is infection. First step is infection. You will get all this PDF on Biology by Gopika Telegram. You can follow me on my Telegram. At the same time, Vedantu NEET English. And students, if you are from Bangalore, I have something very important for all of you. There is a link in the description box. You have a chance to meet all of us on 30th March at JP Nagar Vedantu Center. All you have to do is fill the application, like fill the Google form, so your seats are registered. Okay? Yes. You don't have to write the full name. Just write Hershey and Chase. Bus. Hershey and Chase. Okay, students, first is infection, second is what? Blending. Okay, blending, that is to remove the protein code. Third was centrifugation. This is to get the what is that? Supernatant and pillet. Okay, centrifugation, this is to get the supernatant and the pillet. Okay, very good. Infection, blending, centrifugation. In centrifugation, two results we got. One side we saw the protein was present in the supernatant. Supernatant means what? In a in a centrifuge tube. Okay, can you see it on the white? Centrifuge tube. You will notice that there is some portion which is hard. Okay, like this. Okay, which is heavier. And there will be some portion which is lighter. That lighter portion is called the supernatant. Supernatant, depending on the density. Okay, supernatant. And the what is that? The heavier one is going to be the pillet. Okay, heavier one is going to be the pillet. So obviously genetic material is going to be heavier. Pillet mein we saw DNA, in supernatant we saw protein. Clear to all of you? The entire experiment is clear. Okay, I am from Bangalore. I am from Bangalore itself. Yes. So this is clear. Now students, machinery. Okay, now you should understand that your DNA, okay, one second, your DNA replicates in a semi-conservative manner. Okay, I am not going to get into the depth of it, but all of you know DNA replicates in a semi-conservative manner. That means our DNA is giving us a hybrid, okay, hybrid structure. That means if one heavy, one light, one heavy, one light, one parent, one daughter. That is how we get it. Okay, hybrid means, let's say this is the parent, this is the daughter. Okay, this is the daughter. So we get a hybrid. This also you people know, no? Now to prove that semi-conservative, our DNA replicates in semi-conservative, we did an experiment that is called as Messelson. I mean, not I didn't do, they did, okay? Messelson and Stahl experiment, and this experiment was with N15 higher isotope. Then these people are not radioactive. Please understand, these people are not radioactive, they are just higher isotopes, okay? And we took N14 H4Cl. And we understood that our DNA replicates in a semi-conservative manner, okay? And we did the centrifugation, only important points I am telling, okay? And we did the centrifugation with the help of cesium chloride, okay? Cesium chloride, CSCl. We did gradient, density gradient centrifugation with the help of cesium chloride. These are the important points to remember, and understand here one more thing important is we took E. coli, that is Messelson and Stahl took E. coli because E. coli replicates in every 18 minutes. Sorry, 20 minutes. Okay, in every 20 minutes. So we wanted someone who would replicate faster so that we can do the experiment. So we took E. coli, which will replicate in every 20 minutes. As you are understanding, please put thumbs up. So we understood that we calculated every 20 minutes, then 40 minutes, then 80 minutes, and then 100 minutes. Okay. So this was the calculation. Clear? So in the first 20 minutes, we would have got full hybrid. Right? We would have got full hybrid. Then in next 40 minutes, we would have got light plus hybrid. Okay, light plus hybrid. Hybrid means one heavy, one light. Okay, so we would have got 50% light, we would have got 50% hybrid. Then in next scenario, in 80 minutes, we would have got light plus less hybrid. Why, students? Because N15 will slowly start fading. So we would have got 75% light, 25% hybrid. Okay, next we would have got 12%, 12.5% hybrid, and around 85 or 86.75% we would have got light. Clear? So this is how it happens. So this is the calculation. Now moving forward to the main thing, machinery. Okay, machinery I will not teach you as in depth I teach for NEET students. I will just teach you what is there in your NCERT. So students, obviously if replication has to happen, you know that one has to be the parent DNA, okay, and one has to be the newly formed daughter DNA. Now when parent DNA we want to separate, how do we separate it? For that we need an entire mechanism to happen, right? So we cannot separate the entire 2.2 meter DNA. So what we have to do, we have to separate it slowly, okay, slow. So that separation, that unwinding happens with the help of an enzyme called helicase. Okay, unwinding will happen with the help of helicase. Clear? So here we will form a replication fork. We will form what? A replication fork. That means both of the DNA strands will unwind and they will form a Y-shaped structure, and that is called the replication fork. Okay. Now students understand, for replication to happen, we need ORI site. Even when I teach you plasmid, even when I teach you vector, you have to remember this. We have to have an ORI site, origin of replication. Okay. Now, what is the main job? I have to unwind the DNA and I have to form the new template. Simple as that. So for that, I first took the help of helicase, which is an unwinding enzyme. Helicase slowly started unwinding it. After that, I took the help of DNA polymerase, who is going to form the new template for me. But DNA polymerase will do polymerization only in 5' to 3'. That means, let's imagine DNA polymerase is a hero. He is like, I will only work for 5 lakhs. If you want, okay, 5 lakhs I will start. If it is a small shoot, I will give 3, I'll take 3 lakhs. So he comes from a higher to a lower region, think like that. Polymerization will happen from 5' to 3'. Okay, clear? So now what is going to happen, students? Because this fellow is going to do that, we got a continuous strand in this direction. We got a continuous strand from 3' to 5' direction. Okay, one continuous strand we got, continuous or a leading strand. But since this fellow is very particular about 5' to 3', it was difficult for us to work from 3' to 5'. So what we did, we took the help of primer and we did reverse. Okay, and because of that, we got some fragments. We got some fragments which are called as Okazaki fragments after the scientist. Okay, Okazaki fragments. Since they are broken, we will call them lagging. You are very lagging because this fellow is complete, it is leading. So these are called as lagging strand or discontinuous strand. Clear to all of you? Yes or no? Yes, it is energetically a very, very expensive process as well. Clear? Okay, chalo. Now this is clear. So we got a continuous strand, we got a discontinuous lagging strand. DNA polymerase will do the job of forming the new template. Clear? Now, who is going to do the gluing of the Okazaki fragments? Who is going to glue the discontinuous frag? For that, we will take the help of ligase enzyme, which is called a gluing enzyme. Clear to all of you? This is how much is given in your NCRT. Okay, this is how much is given in your NCRT. They have mentioned about replication fork, origin of replication, DNA polymerase, ligase, helicase, that also not clearly mentioned, and the rest is already on the diagram. Clear to all of you? Any doubts in this? Any doubts in this? Hoping that you are not studying this for the first time. Yes. Now students, coming to the most important, transcription. That is, we understood how replication happens in the central dogma. Now our job is to convert DNA to mRNA. If I want to convert DNA to mRNA, the process is called transcription. The process is called transcription. Yes ma'am, I agree to this. I agree to this. Right. So if the process is called transcription, how does transcription happen? Now comes the question. Transcription has a unit called as transcriptional unit. What unit? Transcriptional unit. This is important for your exam. Okay. Transcriptional unit means, students, three are there. Three transcriptional units are there. First one is called promoter. Now you tell me what will be the job of a promoter. You will get promoted, no? Promoted. Okay, promoter is going to do the initiation part. Then we have structural gene. We have who? Structural gene. Then we have terminator. Terminator. Now, understand, promoter will do the job of initiation. Promoter will do the job of initiation. Yes, ma'am, agreed. Ma'am, what is structural gene? Students, it is a small region on the DNA who will form the mRNA. Imagine that, okay, Rihanna is telling, no ma'am, we need to make this 2.2 meters DNA also into mRNA. How can that happen? So, I will say, no Rihanna, we are going to take a small portion of the DNA, which is called the structural gene, that is going to form the mRNA. Understood? So, structural gene region, elongation will happen. Elongation will happen. And terminator region, termination will happen. Termination will happen. So look here, students, if you look at this diagram, you will understand exactly. So now all of you tell me in the chat box. Now all of you tell me in the chat box, will I use DNA dependent DNA polymerase or will I use RNA polymerase for this process? Come on, quickly in the chat box. Now look here, this is your entire DNA. Now in this DNA, we have taken RNA polymerase. Why? Because you have to ultimately form mRNA, not DNA. You have to form mRNA. Now promoter will do what? Initiation. Yes ma'am, promoter will do initiation. So when this promoter is going to do initiation, it is going to, but students, one problem is that RNA polymerase, no, he only knows one thing, that I have to form mRNA. He does not know anything else. He does not take care of the initiation. He does not take care of the termination. So what we can do? Okay, 1 second, students. Okay, so students, yes, that is, ma'am, we are going to use an RNA polymerase, but the problem with RNA polymerase is it does not know much. It's like, okay, I have come here for one job, to make mRNA, I'll do that and go. But we cannot do that, no, we need initiation, we know when, we need someone who knows when to start, when to stop. So ma'am, are you going to remove RNA polymerase? No, 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 students, I do not remove bad people, no, we do not remove them, we just tell them, wait. So RNA polymerase, we are going to give first a fellow, that is the sigma factor. We will tell RNA polymerase, you do one thing, you bind with sigma factor and you please take care of initiation. Sigma factor will be like, okay, fine, done. It's like a small kid who does not know when to start and stop. So we will be like, okay, sigma factor will come, hold RNA polymerase and start the initiation. RNA polymerase know one thing. What is that? Elongation to form the mRNA. So by then sigma factor will go. RNA will form, RNA polymerase will form newly formed mRNA. Then it does not know when to stop. Then we will be like, okay, don't take tension. We will give you one more person, which is the rho factor. We will give you rho factor. Rho factor will combine with RNA polymerase and termination will happen. Okay, termination will happen. This one, sir. Yeah, we should give them a lunch break. Yeah. Okay, yes. So students, rho factor will combine with RNA polymerase. Sir, 1 second, student. Students, sorry. Okay, we wanted to give you break, no? So we were just discussing about that. Hello. Now this is clear now. So here I told you RNA polymerase is like a small baby. He does not know when to go, where to go. So we will be like, okay, don't worry. We will give you rho factor. So rho factor will make sure that termination happens. So this is the entire thing. So what you have to remember here, there is transcriptional unit, that is promoter, structural gene and terminator, and initiation, elongation and termination is the process that happens in transcription, and during each of the process there is one people who is involved. Here it is sigma factor. Okay, I'll just write S. Here it is rho factor. Clear to all of you? Yes or no? Yes, quickly hit the like button. Students are telling, listen now at least. Okay, your own friends are telling. Now students, most important thing, one more here is, when I talk about mRNA, it's not very easy. Life is not very easy for eukaryotes. It's a little complex. So here what happened, when I got the mRNA, I noticed that this gene is actually a split gene. What gene? Split gene. Okay, the gene that came out is a split gene. Ma'am, what is split gene? Split gene means, students, they have introns plus exons. That means I want some person who is good at sequencing, and I also have one person who is not good at sequencing. A person who will do the sequence, a person who will not do the sequence. Now ma'am, what's the big deal about it? The deal is that, students, these people cannot make the proteins if they are together. I will only be able to make the protein if it is only exon. Okay, if it is exon, I can make the protein because it's a sequence. Okay, it's a sequence. Now this is non-coding sequence. With a non-coding sequence, I cannot make any sequence. No, from the name only you understood. So when I got the split gene, I was confused what happened. Then they were like, don't worry, we have a process which is called as splicing, not slicing. Okay, you're not cutting vegetables. We have a process which is called as splicing. Okay, we have a process which is called as splicing. In this splicing, what we are going to do is, we are going to, with the help of this process of this whole thing is called RNA processing. Okay, RNA processing. This does not happens in prokaryote. This only happens in eukaryote. So students, this whole process is called RNA processing. So during RNA processing, there is a technique called as splicing, with which we will remove the introns because he is non-coding. Nonsense he is. I do not want this fellow because I cannot make protein. So I will remove this non-coding sequence with the help of RNA splicing, and then, because students, problem already happened. So to make sure that there is protection, and since mRNA has to go to the cytoplasm after this, because the newly formed mRNA has to go to the cytoplasm for translation, I will be like, let's do one thing, let's properly protect it with the help of capping and tailing. So we will do here capping with the help of methyl guanosine triphosphate at the 5' end. What is it? Methyl guanosine triphosphate. Okay, should I write it here somewhere? Okay, methyl guanosine triphosphate. Okay, methyl guanosine triphosphate. Okay, so with the help of methyl guanosine triphosphate, I'll do capping. Then you will be like, ma'am, you didn't give tail only 5' and you have given. So I'll be like, okay, fine, let's do adenylation. Okay, what is it called, students? Adenylation, and let's give poly A tail as tailing. Okay, at the tail. So now I have got a newly formed mRNA, which is not a split gene. I've got a newly formed mRNA with proper 5' with proper 3' and with only exons in the middle, which will ultimately code for the protein. Got it? Clear or not? Yes. So this can also, we call this also as HNRNA. Okay, that is HNRNA, heterogeneous nuclear RNA. Okay, this term also is given in your NCERT. Done? Okay, heterogeneous nuclear RNA. Clear? Yes. What is splicing? Removal of introns and joining of the exons. Correct? Done, done, done. So here, students, adenylation, we will add many adenine, A. Okay, A. Yes, this is your RNA processing, which happens only in eukaryotes, not in prokaryotes. Okay, this is happening only in you. Okay, you. Done. Can I go further? Now, students, here in genetic code, don't break your head studying everything. Just study the salient features and go for exam. 1 3-mark question though will come. Okay. So sorry. Drop the pen on my leg. Okay. Yes. Tell me, students, salient features of genetic code are as follows. This is instance, what we call instance. That means naturally, sound will come from our mouth when something bad happens, like something falls or something. No, this is what happened here. Yes. So salient features of genetic code are as follows. Only study the salient features. What is it? Genetic codon is triplet. That means, students, 64 codons were there. In that, 61 code for amino acid. 61 code for amino acid. 3 are stop codons. 3 are stop codons. Got it? Now, some amino acids are coded by more than one codon. Some amino acids are coded by more than one codon. So, we call it degenerate. Okay, codons are degenerate. Clear? Now, the codon is read in the mRNA in a contiguous fashion. That means what? That is, ma'am, if you write AUG, if you write UUU, if you write UAC, I will not read it as UAG, UU, UAC, full stop. I will read it continuously AUG, UUU, UAC. So, it is read in a continuous fashion. Okay. Thank you, Bhusher. The code is nearly universal. That means from bacteria up to humans, UUU will code for phenylalanine only. That means from bacteria up to the highest organisms, code is going to be universal. Okay. Some exceptions are there, but then keeping that exceptions separate, majority are universal. AUG has dual functions. AUG has dual function. What is a dual function? It codes for methionine plus it is a start codon. Start codon means what, students? On the mRNA, this code is going to be there. Right, because we are going to do translation. Translation, why do we use Google Translator? Students, energy is going down. Come on, pick up the energy. I'll give you lunch break, don't worry. Okay, so genetic code is going to be present in your mRNA. We are going to do translation. Why do we use Google Translator when I don't know a language? I tell Google, please type how are you in Tamil. So they will write it in how are you in Tamil and give, right, epidi irkinge. Now that epidi irkinge from how are you, this translation happened because that translator is able to do the job. Here also, codons are going to be present. Reading the codons, they will know which amino acids to bind with. Okay, correct. Now students, this AUG is an initiator codon. If we see AUG only, we will start running. Like on your mark, get set, go. If they tell go only, we will run. No, same way like this. Now UAA, UAG and UGA are stop terminator codon. That means what? Stop codons. If we see them only, we will stop. Okay, if we see the finishing line only, we will stop. No, same like that. Clear? Yes. Now coming to translation. Okay, coming to translation. Ready? Okay, ready? Thank you so much, sports. Okay, translation, students. What is happening in translation? Just like transcription, I'm going to teach you in a very simple way. mRNA to protein. mRNA to protein. Ma'am, but why is it called translation? Just because I told you right now that it is translating. What is getting translated? There is genetic code. There is genetic code on the mRNA, right? There is genetic code on the mRNA. Reading the genetic code, we have to form the polypeptide. Okay, polypeptide chain. This polypeptide chain ultimately is going to see how you will look, what are you going to be like. Okay, polypeptide chain. Your phenotypes will be decided. So, polypeptide. So, this translation has to happen. Now, students, in your textbook, there is one very important thing that is mentioned. Translation, the first step is what? Charging of tRNA. Charging of tRNA, or the word is amino acylation. Amino acylation of tRNA. Amino acylation of tRNA. Ma'am, what is this charging of tRNA? Please remember this, okay? Please remember. Continuous fashion means continuous fashion. Okay, continuous fashion. So, students, charging of tRNA means, okay, what is exactly happening in this image we will see, then I will come back to this, okay? Now, students, all of you have gone in a bus. Anyone who has not gone in a bus, who is that rich in this class? I want to know, okay? Who has not gone in a bus? If you are in a bus, you all know that you will enter the bus. Okay, let's say this translation to happen, there should be two main people. That is your tRNA, who is going to act like an adapter molecule, plus you need ribosomes, who are going to ultimately help us in the formation of the protein. Now, these ribosomes, okay, ribosomes are two, larger subunit and smaller subunit. When the smaller subunit come and bind only, the translation will start. So, this is the larger subunit, this is the smaller subunit, this is the tRNA. Two people are already aligned, okay? Two people are already aligned. Yes, ma'am, two people are already aligned. Thank you. Okay, now this entire thing has three regions. Ribosome has three regions. E site, okay, P site and A site. Now, ma'am, what is this? Now, when you enter the bus, the conductor accepts you, right? Conductor accepts you means what? That means he'll tell, okay, bunny, you know, there's place inside the bus, come inside. So, you enter the bus. Now, you have a bag. Now, let's consider this bag as an anticodon, because your tRNA is going to read the mRNA codon and it's going to make anticodon, and upar se it is, from the top it is going to attach the amino acid. Today, so much Hindi is coming, so much Sanskrit, Hindi, everything is coming. Okay, all languages except English is coming today. Okay, there are days like that, students, in my life. Okay, yes. Okay, yes. So, what is going to happen, students? This, let's say you entered the bus. Okay, you entered the bus, you have a bag in your hand. You are eyeing at the peptidyl site. Peptidyl site means the site where you are supposed to sit. Same way, this tRNA who is coming also wants to go there and join and form the polypeptide chain. So, you are also standing with the bag in your hand and you are looking at the seat when this fellow will get up and go. The moment that fellow gets up and go, you go and sit. Now, exactly that is what is happening here. See here, you entered the bus, you have the anticodon with you, you also have the amino acid which you are supposed to add. You are waiting for the site to where you can join. Can you see how this arrow mark is pointing? Look here, the arrow mark is pointing to add this. The moment this fellow adds this chain, he will move to this side and you will immediately come and sit on that seat. Now, once you add the chain, that means once your stop comes, what will you do? You will exit from the site. That's all translation happens. That means we have to continuously form the polypeptide chain. So, for that, we have to get in, we have to have an anticodon, we have to add the amino acid depending on the anticodon, and then we have to make the polypeptide chain. Is this clear to all of you? Yes or no? Is this clear to all of you? Quickly tell me in the chat box. Quickly tell me in the chat box. Clear? I'm multi-talented. What does it mean? I'm multi-talented means. Okay. So is this clear to all of you? So students, here main thing is, now I'll tell you about what is charging of tRNA. Now what has happened? Let's take an example. Seat is empty, but one old lady is standing, or let's say one child is sitting, and you're annoyed that the child is sitting. So what you will do? You look at the next lady who is sitting in that other seat, and you'll be like, you do like this action. So the moment the child gets up, this lady will keep the bag here and reserve a seat for you. For that, you both should have a connection. You both should have some sync. Same way here also, if two tRNAs are charged, they will immediately form the peptide bond between them. Otherwise, this will not happen. So, charging of tRNA is necessary so that we can form the peptide bonds. We can form the peptide bonds easily so that the whole process can continue until we form the polypeptide chain. Clear? Now, ma'am, how does this terminate, ma'am? How does this come to an end, ma'am? When does this stop? How long they will form this? It will stop when they see a stop codon here. When they see a stop codon, they will stop. But other than that, we have something called as UTRs, untranslated regions, which are present before the start codon. Because if it's present after the start codon, then it will get stopped. No, UTRs are untranslated regions, some regions which are not going through the translation. Why is this present? To increase the efficiency. Okay, UTRs are going to be present before the start codon, and UTRs are going to present after the stop codon. Arrow marks you should notice. This will increase the efficiency of the formation of the polypeptide protein. With that, we also have release factor. Release factor, which will also help in termination. So, how many things have you studied? Is this clear? Any confusion? Anyone has any problem? Quickly tell me. Faster, faster. Dandanada. My throat is drying. My throat shouldn't give up on me, students. Please pray. Done? Clear? Perfect. Okay, moving forward, students. Now, regulation of gene expression, very important in that lack of operon. Regulation of gene expression means what? Full time in the day you will be active, you will be jumping around, oh yeah, okay, you will be jumping that, okay, nakumuka, you will be dancing every time. No, you will not be. Sometimes you will be very moody, sometimes you will be very active, sometimes you will be like, ma'am, I really don't want to do this, right? So, same thing happens in gene also. Sometimes gene will want to express itself, sometimes gene will not want to express itself. So, during this time, how do they switch off and switch on? That is what you have to understand. So, students, in prokaryotes, at one stage, only at one stage, we can do this kind of, switching on and switching off, not eukaryotes, prokaryotes, okay. Prokaryotes, gene switching off, okay, or switching on can happen only in the transcriptional, because transcription and translation is coupled. So, only during this situation, the gene can decide to switch off or switch on. Right, but in eukaryotes at 4 steps, gene can switch on and switch off. That is, first step is when the DNA becomes mRNA. That is, students, during the transcription. Second, during the translation. Third, when the mRNA goes to the cytoplasm. Okay, third. Then from cytoplasm to protein. That is translation, right? So, here when DNA becomes, that is during the transcription, this is RNA processing. So, sorry. Okay, first, student, this is transcription, this is RNA processing, then this is when the mRNA goes to the cytoplasm, and this is when mRNA goes to the cytoplasm, and this is translation. Clear? During these 4 situations, okay, exactly, no fear when I am here, okay, when these four situations, gene has decided to switch on or switch off. Like you people only. Ma'am, the internet is there, I am happy. Ma'am, my friend is online, I am happy. Ma'am, Instagram something is happening, I am happy. Like that. No, you decide whenever you want to be mood off or moody or whatever. Okay. Now, understand. Okay. Now, look here. Prokaryotes only in one stage. Listen to me carefully. Open all the ears. Okay. Transcription and translation is coupled. There is no much difference. Okay, so only during this situation, prokaryotes gene can switch off or switch on. Okay, that is when your mom and dad is there, you have to be active. There is no other choice. Like that only for these people also. Only in one situation, when mom and dad is not there, only they can be inactive. Here, 4 situations they can be inactive and active. That is transcription, RNA processing, travel of mRNA from nucleus to the cytoplasm, and translation. Now, luckily, you have to study about prokaryotes, that is, you study about Lac-Operon model, which is done by Jacob and Monod. So, when you are writing about Lac-Operon model, you have to start writing that Jacob and Monod did this. Okay, you have to respect them. Chumma, you can't tell. Okay. Thank you, Subra. Thank you, Ashvika. I think after I reads the name, she will run away from the class. Okay, she'll be like, this ma'am spoiled my name. Okay, come on, Lac-Operon. This is the last concept that I'll be teaching you from this chapter. HGP and DNA fingerprinting you have to study by yourself. You can watch my lecture also, because students, it will take a lot of time. DNA fingerprinting I'll teach you, but HGP I will not teach you now, because HGP is all about by hearting. You just have to mug up and go. That is how the situation is. Okay, now Lac-Operon. Wherever you have to put understanding, I'll teach you. Lac-Operon concept, what happens, students? Gene has to be switched on and switched off. Ma'am, depending on what, ma'am? I am telling you, depending on the presence of lactose. This fellow is the hero here. Now, what do I mean? I'll tell you. Now, let's take example, RNA. This is in prokaryotes. In prokaryotes, only one situation that can switch off and switch on, that is transcription. So, I will use who? RNA polymerase. So, RNA polymerase is coming. Okay. But what has happened, students? There is one guy, okay, there is one guy who is called Raju. Okay, Raju is from I house. Okay, I house means from the, so we have Pippo ZYA. How do you remember this? Pippo ZYA. Pippo ZYA. So from the I gene, okay, from the I gene, repressor will be formed. No. So Raju comes from I gene, repressor mRNA, then it will form repressor. Raju is very big. Raju will do what? Raju will go and sit near the operator. Raju will go and sit near the operator. Now, Papa RNA polymerase is trying to come to do the transcription, but as he sees Raju, he will get discouraged. He will be like, you Raju is sitting, I can't fight with Raju, I am one thin person, I can't fight with Raju, so I will go back. So, here what will happen, students? Transcription won't happen. Right? Transcription won't happen. Why? Because repressor is sitting on the operator, repressor is blocking the operator. So, transcription will not happen, that means gene is switched off. Gene is switched off. All this happened, why? Because the hero went to abroad. Okay, hero went to abroad. Now suddenly hero comes back to the village. Okay, hero comes back to the village. Everyone is talking about Raju. Raju means R for repressor. Okay. Now everyone is talking about Raju. Everyone is scared of Raju, especially RNA polymerase. And RNA polymerase is like, I have to do the gene switch on, otherwise transcription won't happen. So then repressor will, that is your hero will come. Hero is going to be who? Inducer. So again repressor mRNA, that is Raju, will come out of his house. He will be walking towards the operator. Suddenly he will meet the hero. Who is the hero? Lactose. Lactose will go and hold Raju, telling that I will not let you go, and they will make an inactive repressor. That means Raju, this hero will come and tell, hey, if you don't go to the operator, I will give you this chocolate which I bought from foreign. So you sit here only, we both will talk. So this will become inactive. Why? Because lactose and repressor will make a complex which is called an inactive complex, because of which repressor will not go and bind with the operator, and RNA polymerase, who is coming to do transcription, will happily come and do transcription. Now ma'am, what is this ZYA? These people are the structural genes. Z is going to release beta galactosidase, which will break down lactose to galactose and glucose. That is a function of this girl, not this girl, this beta galactosidase. Okay. Then students, permease will increase the permeability of E. coli. We are talking about E. coli, okay. It will increase the permeability of E. coli to absorb more lactose. And trans-acetylase function is not known yet, okay. Clear? Thumbs up if you understood. So, here gene is going to be switched on. Gene is switched on. Why? Because we understood that we got the lac mRNA. We formed the mRNA. What is the transcription job? From DNA to mRNA. We got the mRNA. We all got the mRNA. Okay. So here what is happening? The gene will be switched on because lactose has come. Okay. Because lactose. This is a leaky model. That's why lactose is entered in the starting. Okay. So students, Raju and inducer, that is inducer is your lactose. Okay. Lactose will become friends and they will become inactive, because of which RNA polymerase will go forward and do transcription. We will get lac mRNA. Again, ZYA gene are called as the repressor gene, structural genes. They will release beta galactosidase, permease and trans-acetylase. Okay. Do you speak Tamil, ma'am? Ma'am, E. coli is chumma sitting, so we thought we'll take E. coli. Okay. Yes, this is clear. So in absence of inducer, the gene will be switched off. In presence of inducer, the gene will be switched on. Clear? Yes. Now, goals of HGB students, this is important, that's why I have put it here so that when I give you the PDF, you study from it, okay. This also same thing, you have to study this, at least few points you have to study, okay. Now, here students, this is very important. Chromosome 1 has most genes, chromosome Y has least genes. MCQ can come. Now, DNA fingerprinting, very important. DNA fingerprinting, what is that? 99.9% of the DNA is same. Can you believe Gopika ma'am and you are the same? How much? 99.9%. So, this is called as the bulk DNA. Every human's 99.9% is the same, bulk DNA. But ma'am, what makes you and me so different? 0.1%. This 0.1% DNA is different, and because of that only we are able to do DNA fingerprinting. Let's say one criminal is there, mine and his DNA is same, then how will I do DNA fingerprinting? They'll put me into police station saying that, oh, you are there, chumma sitting and taking class, so I took you and came. Okay, because criminal would have gone somewhere else. No, I am sitting and doing classes. So to calculate, to find that who is individually different from each other, we have to understand that 0.1%, okay, DNA fingerprinting. Okay, HGB, please study from my previous video here, because HGB will take a lot of time. If I take this much time, after some time you will start crying, ma'am, how long are you taking class? I have evolution, biotechnology, human health. Not possible, okay. So DNA fingerprinting. DNA fingerprinting happens because of this 0.1%. These are called as repetitive sequence or satellite DNA. Repetitive sequence or satellite DNA, understood? Now these students, they have something called as short tandem repeats and VNTR. You please study VNTR because it's mentioned in your NCRT. For CBSE, no need so much. That is called as variable tandem repeats. Now, what makes you and me different is because of this variable tandem repeats. The variability is there because of which we are different. Clear? Now, mini satellite and all that is not needed. That is for NEET students. Okay? That is for NEET students. Now, look here. The steps involved in DNA fingerprinting can come as a 5-mark question. What is exactly happening? First, you do extraction. Okay? First, you do extraction. That is, you extract it, and after that, you use restriction enzyme. This again, detail you will study in principles. Okay, you will use restriction enzyme to cut, okay, because restriction enzymes are called as molecular scissors. After cutting them, you will take the DNA samples and then you will run a gel electrophoresis. Why do you run a gel electrophoresis? Because you want to scatter the DNA depending on their size. You want to separate the DNA depending on their size. After separating them, you will transfer it to a membrane, that is, you call it southern blotting. You will do southern blotting. Southern blotting. And then students, you do incubation with labeled probes. This is important. These probes that you are using is called as VNTR probes. Ma'am, what are they going to do? Now let's take example, okay. Let's take example. Basavaraj sir stole the pen from the studio, okay. Basavaraj sir stole the pen from the studio. So I want to find who is the person who stole the pen. So with the help of fingerprint, I will try to find it. So now let's say in Basavaraj sir's body, okay, in Basavaraj sir's gene, ATGAATTC. This is repeated 50,000 times. 50,000 times. Okay. Now, how do I find that if this is the correct one? Yes, ma'am, according to the size you separated it. But I will make a VNTR probe that is complementary to this, so that in the auto radiography, I will be able to 100% confirm that Basavaraj sir stole it. Okay. So, I will make TACTAAG. This is my probe. This is radioactively labeled. Now what will it do, students? It will go correctly bind to this repeating unit. Because I have so many people samples. No, I have so many teacher samples. So in that, I know that Basavaraj is this one. So if this goes and binds, and if I see it in the auto radiography, I will confirm that Basavaraj stole the pen and went. Okay. So for that, we use a VNTR probe, variable tandem, number of tandem repeats, which are radioactively labeled, and after that I will do an X-ray or I will do an auto radiography. Because it is labeled, I will only be able to see it under that auto radiography, and I will be able to understand where did this fellow. Can you see the binding? Can you see how nicely it is highlighted? So all these places your VNTR probe went and bound. Done, 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 done. Understood? Yes or no? This again detail you will study in your biotechnology. Some you will study in your biotechnology. Perfect. So here are the steps. Isolation of DNA, digestion of DNA by restriction endonuclease, separation of DNA fragments by gel electrophoresis, transferring with the help of southern blotting, western blotting, eastern blotting is there, but here we can use southern blotting with the help of nitrocellulose or nylon membrane, hybridization using labeled VNTR probe. That means you will go and tell, hey, VNTR probe, go and attach. After that, we will use it for autoradiography to see if it is pakka or not, okay. Otherwise, again, they will catch me and go. So that's it. Done, done, done. Molecular basis done. Next is evolution. After break. After break. Because A ray wouldn't look nice, B ray wouldn't look nice. That's why. Okay, 1 second, students. Let me ask Basavaraj sir how long to give you break. Students, we also have to have lunch, so the break will be little longer. It will be a proper lunch break. Okay, because we don't have lunch, no, we have to eat lunch. Yes? So we will take a lunch break now, because if not now, then later the class will become too long. Okay, then I have evolution, biotechnology, principles, application. Okay, understood the whole chapter, molecular basis, because I know most of you are scared of the chapter. Don't be scared, it's an easy chapter. Okay, don't be scared of the chapter, nothing to be scared of. By fear, Gopika ma'am is here. Okay, lunch break. What time, yeah? It's almost 2. I think we need to give up to 25 minutes. Let's see, students, in case the food doesn't come, I'll drop you a message in the chat. Okay. After that, evolution, we'll do fast. Done? Yes, ma'am, understood. Very clear. Thank you. Quickly smash the like button also. Okay, I'll put the target as well. Otherwise, this confused people will not do. Can you see orange? Okay. So 25 minutes. Yeah, I told them.